I hope everyone got a good night's sleep. It's morning for you, it's evening for us, and I really am enjoying spending time with you. And again, it's an honor and a privilege to work with you and to do CRLI in the inaugural Australian presentation. And we look forward to your comments, by the way. Rick and I would love to hear how we could, if there's things that we can do to make it better and to continue to partner with MND Australia. We really appreciate the opportunity. So, and I think I'd like to get some of the lecturers I heard yesterday. I think we need to have them work with us over in the US when we do ours, because we're doing some Zoom CRLIs too. What do you think, Rick? Rick just came back online.
Yeah, no, I'm still here. I just took myself off to not be a distraction, but yeah, no, those were fantastic.
So
really interesting what's going on over there.
Yeah, no, I think it's important that sharing, that's how we're going to get collaborations, not only within our own countries, but across the globe. And that's how we're going to make progress and go further if we all work together. But now we're going to move into what can be daunting to folks who aren't used to reading research papers. But to be a good research advocate or a good advocate, you want to be able to read the literature. You saw a few ways that the headlines may be misrepresenting data until you really get into it, and the headlines misrepresent what's really in a paper sometimes. And so we're going to go through how you might read a paper, go through it, and then we have an exercise, some questions about the stem cell paper that you all should have received, that we'll go into groups to work on, and then we'll all come together and go through the questions. So, but the first part of this section is really to go over how do you read a research paper. And I'm going to try to share the screen. I think, let's see, is that correct? Okay, are you seeing how to read a research paper? Is that what everybody's seeing? That good, okay. So, we're going to talk about how you read a research paper, and you don't have to be a scientist to be able to think critically, right? We can all be critical thinkers. And let's see here, move it along. Oh, there, I got to do it here. So, this is an outline of what we're going to talk about. We're going to talk about some general pointers first, just sort of an overview, and you'll hear them repeated through the talk just to kind of solidify it. We're going to talk about what are the sections of a research paper, and then how you go through a paper step by step. And so some of the pointers: think critically. Don't assume that the authors are correct or they know more than anyone else. That you think critically. It's okay to ask questions, and you want to ask, when you read a paper, are the investigators solving the right problem? Did they set the stage with their background, and did it make sense what they're doing? Are they approaching the problem logically and making reasonable assumptions? You'll think maybe, gee, that isn't the way I would have done it. And they say, oh no, but they know what they're doing. Well, maybe they don't. So it's okay to question and read creatively. What do you think are the good ideas in a paper? And can you translate that elsewhere? You want to take notes, right? Include questions and quick criticisms right in the corners, underline, when I was in school, well, I guess we learned throughout our lives, but when I went to college and medical school, I don't think there was a book I had that wasn't like overlined in yellow highlighter almost completely, to the point where I couldn't resell them and make some money, But read carefully, underline the key points, mark what you think is most important, or what you think has the most questionable data, or something that's not as believable. And once you read through something, you should be able to summarize it in a few sentences. And when you do that, that helps to really solidify, what you read, and may point out to you what you want to go back and look at again and reread. You can consider looking at the references and the background to compare the results of others in the field. So, if you go back into the references, you can read what this work was predicated on. And really important, I think, is what did this study add to what we know? How did it grow the literature base? And, it's going to be slow at first, but over time, it's going to get easier and faster. So let's talk about what are the sections of a research paper. And we'll go through what each section is. There's an abstract, an introduction. Then the introduction should introduce the question, lead right into the methods. How are you going to answer the question? And what were the results of, the experiment you did or the research that you did? And what do you conclude from it? How does it fit into the context of what's known? And where do we go from here? And then the references so that you can go back and look at what the research was predicated on. So let's start with the abstract. That should be a summary of the paper. It highlights the main question, a little bit of background, tells you the key results, and then gives you an overview of the conclusion. It's really the summary of the paper. The introduction is really important because it sets the stage. It should give you a little bit of background, what's been done in the field and why, and it should really lead directly to the question being asked and outline the basis for the study and the question. That's followed by the methods, which is how did you design the study to answer the question? How are you going to carry out the experiments? And then the results. Well, what did you find? What was the data? And then the conclusions. Well, I got these results. How do I put them together? How do I put them together in the context of what's been done? And how do they add to the field? And what's the next big question that comes up? And of course, as I mentioned, the references, which is really the literature. So when we go step by step, so now you know the parts of a paper, you've got some key ways to read a paper. Let's do a step by step. So when you read, look up the words you don't understand. Google and Wikipedia can be your friend. It's true. I don't want you to be in Dr. Google, and Wikipedia isn't always right, but it's a starting point to know what a word means. And very often if you Google a word, you'll get that little paragraph pops up that gives you the definition right there and then. Take notes and go back and reread if you're not sure of something. Start with the introduction. Everybody's tempted to read the abstract first. Don't. Start with the introduction. It should present that groundwork, as I said, and really, when you're done the introduction, you should be asking the same question. It should lead you to ask the same questions the investigator is asking. It should identify what the big question, what the problem is that is to be solved. And you should walk right into why the investigator is doing what he's doing. And you want to think about it critically. Here's the critical part. Did this make sense? Is what the investigator doing making sense? Would you do the same thing? And make sure before you move on that you can summarize the background to the question and what it's going to add to the field. Then read the methods. Identify how they approached the topic. Consider the design. Is it likely to give valid results? I mean, you had a whole lecture on probability and statistics, and that goes into the design of the study and whether or not it's going to yield valid results that can be analyzed. You can make a diagram of the experiments, or if it's a trial, a diagram that sort of outlines the trial. And in the case of clinical trials, I think it's important because that's what I know that a lot of our research advocates are going to be most interested in trials. What are some of the areas that you need to really focus on? We want to look at the population. The study population which receives a drug or an intervention, and the controls. Were they matched? How was the intervention administered? Was it a medicine or some other, a BiPAP or some other intervention? How was it applied? Did they look at safety? How did you choose the treatment and control groups? Did you do it randomly or not? I mean, best design is random. You want to reduce bias, right? It's not, well, we'll put all the tall people over here and all the short people in the control group. We'll put, whatever. You want it to be random. And how did they measure the effect? What were the endpoints? And how did they analyze the results? And that's where that whole lecture on statistics and probability and bias yesterday came in. And then read the results. Now, here's most of the results, the important ones, are going to be in the figures and tables. You can go look at those and read the legends. That's going to be the take home. And when you look at the results, okay, this is the question that was being asked. Did these results answer the question? And in clinical trials, when you look at the results, were the control and treatment groups similar? What safety issues happened? Did the intervention work? And was there significance? Remember that you learned about significance and the probability that something would happen strictly by chance. Now go and read your conclusion, discussion, interpretation. Would you agree with the way the investigator interprets? Did he interpret the results? Did he put it in the context of everything that was known? Can you think of any other way that you might interpret the findings? And, it's really important to talk about, we call it limitations or weaknesses. What were the limitations? Because you need to know that. It's not everything's coming up roses. There may be some weaknesses, and we have to acknowledge them. And does the investigator put the results in the context of what's known and what's gone before? And how did this study help us? What did it add to what we already knew? And what do you do next? Then you can look at the references and you can think about, hey, should I go back and read some of these? This is interesting to me. I want to read the background more. And now go back and read the abstract and see if it accurately summarizes what you just read. Okay, so pretty simple. So don't be afraid to jump in and read the literature. Be an informed consumer. Learn to be critical and creative and ask questions. And with practice, it will be easier. So, I'm going to stop there and we can, let's see, stop my share. We can go ahead and go into our groups and do the questions. And everybody should have, you all have the questions? Yeah, okay. So I'll stop the share, and then if there's any questions. Very good. So Laura, I think you're going to break us into small groups now to go through the questions.
Yep, I'm just assigning people to a group now, so we'll just go in half again. So I'll send half of you off to a breakout room and keep half of you in here. So I'm nearly ready to go on that. How many have I got? 1, 2, 3, 4, 5, 6. I'll just put a few more in. All right.
And
off we go. Yep.
could you share the paper and the questions in your room, Laura, maybe? Just have them up
Yes,
there.
I can. Yep, definitely. All right, off we go.
Laura, do you want me to share my screen for the questions?
Oh, if you've got them there, yeah, we've still got too many people in here. Oh, no, there we go. That's better. No, we've just lost a few more, so that's good. I think we're about even now. Yeah, if you've got that ready to go, Terry, I was just opening it up, but if you've got it there, I'll let you run with that and share your screen if that's okay. Great, thanks, Terry.
Yeah, no, I think I do. It's like, you never know what screen's going to share, right? And so it's even, I even have, where we can find some of this. Did everybody have an opportunity to look at the questions in the paper?
Yep.
Yeah. So, should we go through these? We'll go through all the answers in a minute when everybody's back together again, but let's sort of go through these ourselves. So people can unmute and just, how many people were enrolled? Did anyone, where do you?
That was a bit ambiguous because in different parts of the study and in the abstract, they contradicted themselves.
Exactly. Yeah.
It was like trying to make your way through a snowstorm.
I thought it was just because I was reading it late at night, so I'm glad you said that.
Right. So, there was actually like 23 or something like that. But they had screened 32 or I forget the exact number, but you're absolutely right, Michael. And how long was the trial?
That was contradictory too. He contradicted himself. They contradicted themselves in 3 different paragraphs.
Yes, they did. But it was supposed to be at 12 months. Is what it ended up. Okay, now this is really important. Remember when I was talking about when you're reading a paper that presents a clinical trial, some of the important things, how did, what was the placebo control group? And for those of you who are just looking, if you have the paper in front of you, you can go to the methods section where they talk about in paragraph 3. And so what was the placebo group?
The ones that weren't specifically selected. The
People
ones that
who
weren't
had
the specific
surgery.
10. Everybody else.
Yeah, and was there a sham surgery, like, something that would make you unbiased when you No. And were the investigators blind to the treatment? We talk about double blind, right?
Definitely not.
About double blind, right? Were the investigators blind to treatment? What do you think?
I got the impression they were not blind. I thought they could see everything.
Yeah, exactly, Keith. They were not blinded. And how did they measure safety and efficacy? Were there safety measures?
Well, they did exclude some people, and they did include others specifically. But then I think they crossed their fingers and hoped.
Yeah, but and how did they look at efficacy? What did they measure, if anything?
They measured survival time, and they measured the ALS, FRS scores.
Right, and vital capacity, I think. I don't have the paper in front of me, but
that's right. They measured the VLS.
Right. So okay, and so, but was there any plan for safety?
Terry, I thought the limitation of the number of patients that were treated, so that was set at 10 specifically by the Ethics and Research Committee, and they had specified that that their treated patients would be closely monitored throughout. So there was reference to that, but no specific.
How? I mean,
when I had a look at it,
Did they measure CBC 20, you know, check some blood work, check a CAT scan of the head? You know,
I thought they excluded the people with bulbar offset, but later on they mentioned that they actually used them within the study.
yeah, well, okay, so if you look at, you know, under methods and, you know, we're going through all the answers and we're going to go through them again in a moment when I go through this with the whole group, but, you know, you can see the inclusion and exclusion of, you know, we won't go through those, but they had that was paragraph 2 of the methods where they talk about their study subjects. And now we're getting back into, you know, we're talking a little bit, it's been mentioned, how the treatment and the control group were chosen. You know, do you think that was random selection the way it's supposed to be? Remember?
My observation said that whether they tried to be random or not, there seemed to be a bias in the outcome. The people who received the treatment had much longer from diagnosis to that point than the people who were in the untreated group.
Exactly. What do you think that might have meant?
What it meant to me was that they were setting up to get the result they wanted, because you'll see in the data, the table, that some of the people who were in the non-treated group didn't even live as long as some of the people had already lived before the trial of those treated, which meant that they put the treatment on people who somehow had an underlying slower progression of the disease or had already lasted a lot longer. So they were set up to get success from the way they chose the people one way or the other.
And
Yeah,
another,
so,
I wonder, in fact, sure, they chose the first 10, but I'm wondering whether, as you mentioned yesterday, there were very few people available to participate. So maybe they were limited to this group because of the lack of ability of people who wanted to participate.
well, if you read carefully, Michael, people who, you know, the controls were people who said, I don't want that surgery. So, you know,
Yeah,
and
I
this
noticed
was
that.
actually in Mexico, by the way. I think that's where this study was done. You know, but the point is, as Keith is making and you've made, is that, and I think even Jenny's made, that this was not randomly selected.
No.
So you were, you know, and Keith pointed out, you know, the next question, which is the duration of ALS from diagnosis to baseline was, you know, longer. They were slower progressors. You can even look at the slope of the ALS FRS score. It was much slower in this group. So, you know, it was a pre-select. I don't know that they purposely did that or not, but it ended up being that way. And so you were not comparing a real control group. And, you know, and then we talked a little bit about safety. How many people died in that perioperative period?
They didn't say, except there was a comment that one person, 2 weeks, within a couple of weeks of cardiac arrest. They didn't make a direct connection to the surgery.
Yeah, but typically what we do, Keith, is in a clinical trial, if someone dies that close to a procedure, you know, you certainly the chances are that was related to the surgery, and certainly is a major adverse event. And if they were a slow progressor, right, well, it could have had some intercurrent problem, but, you know, could have had a heart attack, like he would have had a heart attack whether he was operated on or not. But, you know, one of the people in the transplantation group died within that very early perioperative period.
And if we add the one who had respiratory insufficiency, that's 20% of the test group.
Yeah, but, you know, fair is fair. You don't know, these are people with ALS, these are things that can happen, but they both happened in the control group, and you would like a little more information about the folks who did poorly, right? So how would you improve the study design?
Would you increase the number of participants?
Well, you could do that, Casey, but there are other things you might want to do too.
I thought the first thing they'd have to do was somehow get a treated and not treated group or control group that was significantly more homogeneous in symptoms, duration, and the like than they ended up.
That's right. You want apples and apples, not apples and oranges. You know, that would, you know, in a phase 1 type trial, you know, you could think of it this way. You still want the groups to match. For phase 2, if you got a control group, you want it to be a control group. You might, although this is a little bit harder when it's a surgical procedure, but you might want to do sham surgery. You might want to add some clear safety measures, right? Those are all things you could do to improve this. So, you know, overall, what did you think of this as a study?
Not much.
Yeah, but what would the, Keith, what would the headline say based on the abstract?
It's an extension of what I said earlier. It appears one way or another that the selection for the treatment and the selection of the control group predisposed them to coming up with a thumbs up for the surgery. And the data that they collected seemed to be pre-shaped by the way the thing was set up. So that's what it looked like to me.
Yeah, but what do you think the headline, you know, what's going to be the headline that's announced? How about surgery
headline-
prolonged survival?
Yeah, the headline from the
Cells prolonged survival, right? That's going to be the banner. And that's what we were, you know, the statistician was trying to teach you how to look at a paper, right? And read it. And that's what I'm saying. If you read critically, you went back and you looked at this paper and you said, oh my God, right? This is not good.
I would, as a reviewer of papers, I would have rejected the paper, so the press wouldn't have known about it.
Yeah, well, here's the thing. This was published in, I think, Stem Cell Research. And, you know, that it may be that, you know, sometimes you have to get these things out, but you're right. You know, it maybe should have been rejected, but it wasn't. And if you're a lay person out there, and you just read that abstract, right, without being real critical, or somebody blasts out nowadays on social media. You know, the universities are always blasting stuff out. They could blast out on social media. You know, stem cell research shows effect on ALS, slows down disease. And pretty soon, I got a whole line of folks at my door saying, can I get that surgery? You know, and can I get those stem cells, even if they have to do surgery to put them in? You know, this was stem cells. So, you know, it shows you why you have to read the whole paper too. Because if you just read the abstract, you might get excited and want that, want those stem cells. So, you know, hopefully everybody feels like, you know, they're a better consumer of how to read papers now. And
was I being critical in recommending? I mean, I've rejected better papers than this within my own field, you know, as part of a committee reviewing papers for journals. But was I being overcritical?
what, I'm sorry, what about more?
Was I being overcritical in as a potential reviewer in rejecting the paper?
No, I mean, I don't know who reviewed the paper to begin with, but somebody may have felt, well, you know, it's flawed, but it should get out. People, maybe somebody will redo it and do it right, and we'll see if they really work. You know, I don't know what was in the minds of the reviewers. I think it's important to be, you know, a critical reviewer. I mean, I review papers, and it sounds like in your field you've reviewed papers, Mike. What did you do?
I often made, well, in many of them, it was made suggestions for some editing work to be done, and and things like that, accepted with revisions. And
Oh,
sometimes
yeah, well,
with some really bad papers, it was a matter of suggesting that it be rejected. But
Yeah.
this is in a totally different field to this.
yeah, no, what field were you in, Michael?
I was in IT and also in adult education.
Excellent.
So I was reviewing for journals like HERDSA, which is the Higher Education Research and Development Association. That's for the adult education and university stuff.
So maybe, for our group here, maybe you'd like to add some comments about how to read critically and look for things above and beyond what, I reviewed for everybody.
I think your slides are terrific. And what you suggested was really good value.
Okay. Keith, did you want to comment on anything?
The thing that's been tickling my mind since I read it a couple of nights ago is that there's hollow in the way the trial was done, but you'd have to wonder about which entity body approval thing allowed them to have the selection criteria they had in the first place that set the thing up. It seems that not just the team that did the review, you'd have to be worried about the environment within which they were allowed to do it from the start, perhaps.
Yeah, the IRB.
Sorry?
The IRB, the Institutional Research Board at their institution. And, we have the ethics board.
Yeah, and it just looks to me the way the thing read that the starting point for the trial predisposed the direction of the trial. So why it did that, you'd have to be skeptical and go and have a look.
Reading between the lines, my thought is that it's so competitive. It's the publish or perish thing. If we don't get papers out, we lose our jobs.
Yeah, and that actually perpetuates people rushing things, people publishing. I mean, we all understand that. Jenny, did you have any comments you wanted to make?
I wondered whether or not this particular study came about through serendipity almost, that somebody had an idea, they floated it by a few neurologists, and it seemed like it could almost have been opt-in for people who were in the latter stages, who perhaps were willing to try anything, versus those that were at early stages who were perhaps looking much more broadly at what they might accept. So I can see that it could have been serendipitous, and then they just decided to publish the results off the back of it.
Yeah, maybe because the control group were people that said, no, I don't want that. And they turned out, when you looked at those groups, to be very different. So, Casey, did you have anything you wanted to add?
Not additional to what others have already contributed.
Good. So I hope that everybody feels, well, seems like Michael had to read literature critically anyway before, but I hope those of us who were not as used to reading, research literature feel a little more comfortable with going through it and looking at it. And I don't know, Laura, we can have our coffee clutch here. We can I think we kind of finished our review, and I don't know how the other group is doing.
I'll just message Gethin now, but I think a coffee break's a good idea. Somebody was about to say something, sorry?
Yeah, often when I review these sorts of things, I often take, more than two passes, but I certainly take two basic passes for different purposes. One purpose is to look at the content, and the other purpose is to look at the structure and the logic and the consistency. And because sometimes there's gold hidden within bad presentation. And it's important to look at both, to help new authors improve their communication by some good suggestions from when reviewing. But most of the panels I've been on said, don't make suggestions, just comment on what you observed. and let them work it out.
But
Well,
no, I think,
Casey's just popped her hand up as well. Sorry, Terry, you go and then we'll go to Casey.
yeah, no, I was just going to say, I think most of us sort of are all educators, and you want to help the person do a better job. I mean, it's like when we review a grant, and you often sort of say, this would make it stronger, or I was concerned about this, and did the investigator think about that? Because, I take no joy, having been there myself, in, triaging a grant, or in, rejecting a paper. In fact, I almost cringe when people ask me to review, because I don't want to reject a paper, because I know how much people work on them and how hard it is sometimes. And so I always like to sort of help the person along, especially a young investigator. You want them to be successful. I mean, that's what we want. We want every investigator to be successful, certainly in ALS, because their success means we get closer to answers. So, Casey, did you want to comment on something?
I just had a question. I don't have a research background, but now that, reading through the paper, I sort of see the things that everyone else has pointed out. Do you think that the publication of such a paper in the long term hinders the research narrative of MND, or is it still helpful? Or does it mean that if someone else were to put forward a similar research proposal, which might have a slightly variable, who the participants are, for instance, is it still helpful that it's published?
I think it starts a dialogue. And I think it would stimulate people to reproduce and do it right. The problem is, it's like there was a paper and on lithium. It was a flawed trial like this. It was actually a lithium in animals. And then, Keith, I know you want to say something. I'll just go over this and give you an example of what you're concerned about, Casey. This was a paper in, I think it was PNAS, Proceedings of the National Academy of Sciences, or one of those kinds of journals. Very high prestige journal that looked at lithium, and there were some background reasons why lithium could help ALS in an animal model, that mouse model that everybody uses, the G93A, where it was the only mouse model at the time. It predated C9 and TDP43. And the mice got better, and then they did a very small trial on 20 people with ALS, and there was a remarkable improvement that they said they couldn't not give it to everybody because it was so amazing. Okay, well, you can imagine what that spurred, because lithium could be prescribed. So what it spurred was 2 large placebo-controlled trials, God knows how much they cost, and people just going on lithium, which is not exactly, it can cause thyroid problems, and it can cause other complications. It turned out that by and large, lithium didn't make a difference. Now, we now understand that in conjunction with a particular mutation, that UNC13 gene, which is a modifying gene for ALS, that lithium in people with that genetic change, lithium may actually work. But at the time, it spurred 2 large trials that were millions of dollars to find out that it really didn't work. So, you're right. It can get us into trouble, but it can also spur additional research. Every finding should be reproduced separately. And now you can think critically, right? You read that and you could see that this wasn't so good. So Keith, what did you want to say?
It's a general question. I have a background which has caused me to read complex papers in a completely different world. So I'm pretty confident I can read English. But when I try to read these scientific papers, the form of language, I know it's written by one specialist to be read and reviewed by other specialists in their journals, but the language is so convoluted and so complex, I wonder often whether or not the reviewer or the reader actually gets the same message that was transmitted. Is there a particular reason that, apart from the scientific terminology, which has to be precise, that many of these scientific papers are written in an inordinately complex form of language?
Because I think many many of my colleagues don't write English. I mean, I think that, you know, people think they have to say things complexly when they can just say it straightforward and directly, and it would be more understood, Keith. I think you're absolutely right that sometimes the way things are written doesn't have to be that obscure, obtuse. You're not any smarter if you use bigger words and you're not direct.
Yeah,
Right.
it's just an observation. It would make it, I should think, easier if part of the process of becoming a scientist went towards condensing the language towards the simpler end more than the complex end. I think it'd be useful.
Yeah, well, I always tease my, when I'm taking care of pals, and I'll always say, here's doctor speak, but here's what it really means. And, because, I don't know why people can't talk, in our language, it would be plain English, but it could be plain Spanish, could be plain whatever, but just directly, plainly, and clearly communicate ideas.
Yeah.
You're absolutely right. And, I'm sure that, you had to read, complex papers, and maybe they would be like complex to me. And I'd say, why can't you guys write directly? We're used to this, but you're right. We all need to be direct communicators. Matt, did you want to say something?
Yeah, hi.
Good morning.
Just to respond to Keith's comment, I agree. It's lawyers in Australia are being forced to use what is known as common English, and if they could translate the outcomes of scientific papers using the same approach, that would be great. Problem is, I am a scientist and not so much academic scientist, but a commercial. So I do read the papers, and I have both a chemistry and medical background. So the papers I read, I understand better than most. This case to me was very dangerous because in the hands of someone who can't critically analyze like we have today, and someone who is not a scientist, their takeaway would be that this is a saving grace. And I've seen it with some people in Australia. There's a fellow that has gone to Indonesia or Thailand to have stem cell replacement, and he's plastering it all over the Facebook pages, and I know a lot of people are saying, oh wow, this is an alternative, let's jump on. And his non-scientific reactions are even more dangerous than this paper. So it's not just the papers that are of concern, but social media propagation.
Yeah, and I think, I'm not sure if you were here when we talked about, if you didn't think critically, the headline that was generated on social media or in the news would be stem cells work for ALS, extend life.
Yeah, that's right. I was here for that. Just had a few technical challenges.
Yeah, and you're absolutely right. I mean, I remember when people would go for what was it, to Mexico for God, those poison nuts for cancer, you know, and yeah, and I mean there are people who prey on, you know, nothing makes me angrier than the folks who prey on pals who are, I don't want, you know, let's face it, pals want a cure, they want to get better. And there are charlatans who will prey on pals. They'll say, I've got the cure, you know. That's what spurred Rick Bedlack to start ALS Untangled, is to try to dissect those claims of cures that aren't, you know. So we usually, when we do one of those, like somebody will say, oh, it's cured ALS. I'll say, okay, well, can you please give me the number of people you've treated with ALS and the before and after, and, you know, and I've yet to have anybody provide me data that actually shows a response to these crazy treatments, you know. And there is, I will say there's stem cells that are approved in Korea, by the way, for people who are interested, you know, so that's a real stem cell treatment. And of course, there are several companies who are doing stem cell treatments in a real way, you know, in a trial, in a controlled trial, even with sham controls. So for instance, the neuron trial by, you know, Brainstorm, people actually got bone marrows, right? They got bone marrow. They used the bone marrow to make their own stem, you know, the person's own stem cells and give them back. But even the people in the placebo group had a bone marrow and got those LPs and got the stem cells back, so that would be controlled. And so that you had real, you know, comparable populations. But yeah, no, all of your comments are right on. And, you know, and even, you know, for, you know, Matt, you're scientifically savvy. Mike's done some, you know, reviews, and obviously, you know, in a different field, accomplished, and Keith's has read, you know, complex papers, but you could start from scratch and just using your critical thinking and practice. You can read these papers, and it's not to disappoint you, but it's to teach you how to be an educated consumer and be able to separate the good papers from, you know, the really bad papers, and don't doubt yourself, right? Don't doubt yourself. If you think something's poorly done, it probably is, you know, especially if you have your reasoning behind you. So, you guys are a great group, and I hope you got something out of this. And if nothing else, for the folks who aren't used to reading scientific and complex papers, just that you can read this and you can see what the problems are with these papers, that you know, that it isn't as hard as you maybe were thinking, although it seems like we got some people here who are used to doing it anyway. So Laura, are we ready to go back? And
we are just about ready. Gethin's just messaged and said 2 more minutes, so that's pretty much perfect. But I just wanted to thank everyone for that. That conversation was really helpful, everyone, and especially to Matt with your comments around the common language. And it's something we really struggle with, because I guess in a way we're sort of at M&D Australia or Research Australia. We're in between, in a way, the research and scientists and the people we're trying to communicate with. And we have this constant struggle of trying to interpret the research that we fund in a way that people can actually understand. So Matt, you've given me some really helpful ideas there that I was furiously taking notes on. So a massive thank you for that that input from all of you, in particular you, Matt.
you know, possibly a publication board with pals and researchers together, creating your, you know, news, your newsletters and your publications, because that'll match making sure that you don't lose some of the technical nuances in the way you express something. Because I think that's always the fear, is that if you simplify it too much, the nuances may be lost, or it isn't quite right. It isn't said. It's like every time I talk about C9, you know, the C9 expansion repeat, it's not a mutation, it's an expansion. But people call it the C9 mutation, but I can't bring myself to do that because it's an extra piece of DNA in the C9 gene, the C9ORF72 gene. And so I always call it an expansion, or I always say, oh, it's extra DNA that doesn't belong there. And because that calling it a mutation bothers me, because it's not technically a mutation as we think of it. So, you know, but how do you communicate that in a way that doesn't obscure the meaning of things?
I think, Laura, a good example that you might use is, you know, I work with chemists in laboratories, and I work with people who are non-chemists, and more so I interact closely with geologists. I always say every field, the only difference is the dictionary we use. So I spend a lot of my life explaining chemistry to geologists, and you've got to dumb everything down a little bit to your audience, and I think the same goes between all professions, whether you're a lawyer, an accountant, or a geologist. The language needs to be the same, but slightly different dictionary. And that's how I was able to fairly effectively communicate across all ranges of science-based professions, and the guys in the office who are accountants. And you know, you've just got to adapt your message to meet the end user.
Well, I always like to think, I don't like to think of it as dumbing down. I like to think that if I can't explain things in plain language, I don't know what I'm talking about.
That's a good way of putting it, Terry. I like
correct.
Yeah, and so it's really putting things, not dumbing it down, it's just explaining it in a language that's universal.
Yeah.
That we all understand. To Keith's point, I think that he made earlier, it's just being able to talk in a language that's understandable, but no less complex. You know, you can explain very complex ideas in a clear way. I don't want to simple way is not quite right, because they can be complex, but you can in an understandable way that anybody can then understand. And so, you know, it's kind of like I try to explain that to my residents and the folks I train, that, you know, you have, if you know your stuff, you can explain it to anyone in a way they can understand.
All right, thanks everyone. I'm going to put us back into the main room now. That was a really great discussion.
I guess
Thank
I'll
you.
keep the slides up.
Yes, yeah, that should just remain, Terry.
Thank you, Terry.
No, thank you. And thanks to everybody for contributing to the conversation. But you guys all read the paper, so it made it easy and fast.
And
all right, I think we're all back in the same room now.
Yeah, we're all jumping back in, I think. So I think we're all back. So is it back to you, Terry, just to do a brief wrap up?
Yeah, so we'll go through the questions. Hopefully, your group also were good readers. My group knew everything already. I couldn't teach them anything. They were teaching me. So, how many people enrolled? Well, there were 32 screened, 23 enrolled, and 10 were treated, and 13 were in the control group. And as some people in my group pointed out, it was pretty hard to dissect that out through the paper, but it was in there. It was in the results section. How long was the trial? A year. And was placebo or sugar pill sham surgery used? No, and please just unmute and talk up if you want to comment. In fact, I'm going to slow down. And are there any comments or any questions? We had a lot of discussion in our group about these things, but now we've got everybody in here. So please just unmute and you know, ask questions. That's what we're here for. Was the study blinded? Did the investigator know who was in treatment and who wasn't? Pretty obviously, it was not blinded. You know, the blinded means a double-blind controlled study is when the investigator doesn't know who's on the treatment and who's not. How did they measure safety and efficacy of the stem cell treatment? Well, there was no clear plan to measure safety, and there was no safety data presented. The efficacy was the survival, the ALS score, FVC, and then there was a quality of life scale. And again, does anybody have any comments or questions they wanted to make?
Why was 51%?
I'm sorry? Was there someone wanting to comment on that? No. So inclusion and exclusion criteria. Well, the inclusion criteria were they had ALS, their MRIs were okay, they had an FVC that was measurable and an appropriate nutritional state. They were excluded if they had severe bulbar involvement, inadequate nutrition, tracheostomy or gastrostomy, other systemic disorders, and if they had evidence of MRI abnormalities like a cervical spine problem. Now, how were the treatment and control groups chosen? It was done by the site investigator, and it really wasn't random at all, and controls were kind of self-selected by refusing treatment. And again, I'll just wait a minute, see if anybody wants to make any comments. So now we want to look, let's look at, okay, well, the control and the treatment groups weren't really randomized, but how did they compare? So the duration of ALS from diagnosis to baseline, how did the two compare? Remember I told you those tables are where the action is. So if I look at the control group and I look at the treatment group and I look at these numbers, the time to diagnosis was very different. It was 30 months rather in the treatment group and 14 in the control group, suggesting longer duration and maybe slower progression. And you know, in our group, Keith and the folks all recognized that. And I don't know if you guys in the other group picked up on that, to start, this was a slower group. And what about the ALS FRSR score at baseline? Again, here's our table with the ALS FRS at baseline, and here's the graph in the figure. And so if you look at the ALS FRS of the dark, which is the treatment group, they kind of stay the same over 12 months, whereas the control group, it goes down. It's a more rapidly progressing group. So it's very different. The control group was higher to begin with than the treatment group. And so are the treatment and control groups similar? They're very different, right? So already you're comparing apples and oranges. So, anybody have anything they want to comment on before we move on to the next points? Quiet group here.
I mean, I'd just like to say that, as you pointed out, those groups were very different, fundamentally different, and it would have, they were on very different trajectories. So the treatments, the treatment could have had absolutely nothing to do with it, basically.
Yeah.
The results could have just reflected the trajectories of the two groups. This is what would have happened regardless of what you did.
Exactly. We had a pretty active discussion in our group already, which is why everybody's so quiet. But I hope that in your group, Gethin, you had a similar sort of discussion about this poor control group for comparative. And so there was one person who died in that perioperative period. We don't know for certain whether the surgery had anything to do with it, but still one of the people died. Another one had some respiratory problems, didn't die. So this is the key. Now, you guys, we talked a little bit about how to read a paper, what to look for. How can we improve this study? Well, a true control group, placebo controlled, meaning maybe a sham surgery. And maybe you could have somebody separate do the exam. So the people who do the surgery should not be the same people doing the assessments down the pike, so that they're blinded to whether or not that person had surgery or had stem cells placed. And there should be a random assignment of treatment. Well, I don't feel like having it, so I'm not doing it, right? And of course, the collection of safety measures should be part of any clinical trial. So that would have been helpful. And you need more information about the conduct of the trial, because you want to make sure the trial integrity was there, and you want to make sure that there were no major protocol violations. What was the protocol, and did anything happen that would violate that protocol? So what you want to do, our final lesson, read carefully. Don't take everything at face value. We were laughing because the headline, if you read the abstract, could be "Stem cells slow ALS down." Don't take things at face value that you see on, social media. And I guess TikTok's the new social media. I don't know. I'm too old for this. But, read things carefully. Don't take them at face value. Be informed and educate others. That's what, as ambassadors, that's what you want to do. You want to educate other folks. And I just want to say thank you for being a research ambassador and helping us. And with that, I'll stop and certainly, and comments, questions. I think we went over things pretty, a lot in the preliminary discussion of how to read a paper. Then we looked at a paper that had some flaws, and came back and we're all in the same room now. And so the question is, anybody have any final comments they want to make? We had some discussion about in science, why can't we just say what we mean directly, clearly, and straightforward, not necessarily lacking complexity, but having complexity in a way that's understood by anyone, a common language, instead of our doctors speak, our scientists speak, whatever, let's make things straightforward and simple. And now I hope that everybody will feel comfortable reading a paper and going through it. And
Keith, do
anybody
you have a comment?
Yeah, I have one. The question that is not directly related to this paper. This paper was written by people in Mexico in Spanish language, first off, probably translated later, I guess. But that was done in a context of the ethics and the social structures of that country. How universal are those ethics and social structures between countries in international reviewing of these papers? We judge this paper poorly to the risks and the like that we would see. Are they the same risks and the same limitations that a different society would see on that paper? And how relevant is that question to reading these papers internationally?
I'll take a crack at it, Terry, because I spent a lot of time, looking at treatments that are in other countries with ALS Untangled. And I would say, the amount of oversight and the process of informed consent looks very different in different places. It's pretty rigorous in most, westernized countries, but it looks very different. But what's not different is the principles of science, Keith. And so what I would say the big problem with this paper is not anything that has to do with ethics. It's basic principles of science that were not really followed. And the comment I was going to make, Terry, at the end is, people were saying, well, how did this get published? And so what I would say, if this had been sent to me as a peer reviewer, I actually would not have recommended rejection, because I want to encourage people to publish things like this so the world knows what happened. What I would encourage is a much more rigorous explanation of, exactly what happened as far as safety goes. There was no safety table, but also a long description of, the limitations and why we can't draw a firm conclusion from this study and what's going to be done better next time. So we want people to publish, even if the studies are flawed, but please, at the end, just describe what the flaws were and talk about how the next person can do better.
Yeah, we had a discussion around about how could this paper be published in this format if it's been peer reviewed, but unfortunately peer review is a subjective process. It depends on the reviewers. Not everyone, unfortunately, Rick can't review every paper in the world. So sometimes it goes to perhaps people who aren't as thorough or don't have that right expertise, and then they miss real key defects in a paper, and it's out there. And once it's out there, then there's not so much you can do about it.
I used to try, Gethin, to review everything. And then I realized that you can say no and you can suggest other reviewers. So oftentimes now I say no, but please reach out to Dr. Terry Hyman Patterson at Temple. She really enjoys.
She rubber stamps everything. She just. Richard, you're hiding. Rick just went into hiding. Look.
Are there any more comments? I think sounds like the discussions were pretty good. I think most people got everything out of their systems in the chat. Okay, well, we've got a break now. Just go head off, grab a cup of tea or whatever, coffee, whatever you need, and we'll start the next session at 11:15. But feel free, if you're hanging around, to come back and jump online and have a chat anyway until before then. Just switch your camera on if you're back in the Zoom room, and otherwise we'll be off just a couple of minutes before 11:15 if everyone comes in. So we'll try and keep schedule a bit better today, and then we're going to hear Kathy Blizzard's going to tell us all about MND biology. So, the real backbone of it all. Okay, I'll see you all in a few minutes. Pass you over to Kathy Blizzard, who can introduce herself and take us through. Do you want to share your screen, Catherine?
Great. Yep, I think you can
All over it.
see my children. Okay, thank you very much. Let me just get my slides up. Now, this never works the way I want it to. Can everyone see that one?
That's perfect. All good.
Okay, perfect. Great. All right. Well, thank you. And thank you, Gethin, for inviting me to speak today. I always enjoy speaking about M&D research. And so I was really excited at the invitation. I was a little bit nervous when I got my title because that's a pretty big title to be dealing with in the preclinical research space. But I had a thought about what I could talk to you guys about today. And so I just, before I start, I guess I should introduce myself. I'm Kathy Blizzard. I'm a senior research fellow at the University of Tasmania. I call myself a neurobiologist, but I go by a lot of different names, but basically I'm in the preclinical, the discovery research space of motor neuron disease. And so I'm a large team. I co-lead this large team with Professor Tracy Dixon, who you can see there. Can you see my mouse when I point? Does that work? Yep, great. And so we run a large team that is focused on working out what goes wrong in motor neuron disease. How can we understand the molecular biology behind the neurodegeneration that occurs in this disease, and how can we identify new therapeutic targets and more effective ways to treat this disease? So today I'm going to tell you about a little bit about our understanding of the mechanisms behind motor neuron disease. And then I'm going to give you a bit of a flavour of the kind of research that I do. I want you to take away from this that, that there's still a lot of unknowns out there, and I want to sort of bring you to the cutting edge of research. So I'll show you some sort of really exciting and novel data, but just please bear in mind that this is one aspect of a very, very complex disease. So we certainly don't have the answers, and there's lots of really valid and important research going on around Australia and around the world. So this is just a little bit of a snippet, but I will start with a bit bigger picture for you. So when going through my slides, I would obviously don't need to have an impact of MND in Australia slide for this group, but I still decided to keep this slide in here for one reason, because it's important to talk about, but it also, I really wanted to highlight to everyone in this room just how important motor neuron disease is to MND researchers. We're really, I think, in my opinion, we're a really collaborative group of researchers, and that we work really well together, not only in Australia but around the world. And the thing that really brings us together is that MND is really close to all our hearts. You know, in Australia there's 2,000 people currently living with it, and that this is a little bit dated, but you know, it's a huge cost on the economy, but the cost on the economy is nothing compared to what the cost on the person with MND and their family has. You know, this is a really nasty disease. It gets called a lot of different things and a lot of not very positive names, and it earns them all. And you know, just one thing that always kind of stays with me and I think about a lot is, you know, the physical care that a patient needs. You know, that's so hard on them, but it's also incredibly hard on their family. So this affects everyone, this disease. And so we all, you know, all MND researchers really hold this sort of information close to our hearts, and we are really all banding together to try and work out what is going on and how we can stop this. So when I think about motor neuron disease, I think about how does it start and how do we treat it? And so to think about that, we have to think about the system that it impacts. And this is the corticomotor system. Sorry, before I go on, I'd just like to say I'm more than welcome, happy for you guys to ask questions. I'm not very good at checking the chat. Maybe Gethin, if you see something in the chat, give me a yell. But if anyone wants to ask any questions, please jump in. I'm happy to, you know, and I'm more than happy to, you know, if anything needs to be clarified, let's stop and have a chat as we go along. Anyway, so the corticomotor system. So this is what, this is the system that degenerates, stops working in motor neuron disease. It's comprised of the motor cortex, which is part of the brain, as you can see up here, and the spinal cord, and also the brainstem. And the spinal cord, these have lower motor neurons inside the spinal cord, and these lower motor neurons send their axons out to muscle here. And this muscle is innervated by these lower motor neurons, and a stimulation from this lower motor neuron, or a firing as we call it, electrical impulse, enables the muscle to contract, and that enables voluntary movement. Now, this system and these neurons are actually really incredibly vulnerable, as we know in motor neuron disease, and they're a really interesting neuron type. When you think about a lower motor neuron, for example, the size of this in an adult human is about 50 microns in width, the size of this cell body here. So that's so small you wouldn't be able to see it without a microscope. However, its axon, the thing that connects the cell body to the muscle, we kind of think of this as a train track that sends all the information, all the energy down to the muscle. In a 6-foot person, that can be up to 2 meters long. So these are a really crazy cell type when you think about it. You know, 2 meters you can easily see with your eye. It's taller than a lot of people. But yet the cell body, the thing that is kind of the control system of this whole neuron, is 50 microns in diameter. So these cells are really vulnerable. You know, they live in the spinal cord, but there's lots of places out in your body that they can be injured or insulted, and this can, you know, cause a disconnection from the muscle. So these cell types, we have lower motor neurons and upper motor neurons. They're the cell types that we need to protect in motor neuron disease, because they're the ones that degenerate and the ones that stop working. So amyotrophic lateral sclerosis, ALS, motor neuron disease, we use these terms really interchangeably. In America, you might be able to correct me if I'm wrong, but I still think some people also still know it as Lou Gehrig's disease. So there's many different terms for this. And I just wanted to say that, you know, ALS is the most common form of motor neuron disease. And so that's why we use these terms interchangeably. But this is the, when we're talking about motor neuron disease, and when I'm talking about motor neuron disease today, what I'm actually talking about is ALS. And this is the one that leads to the progressive paralysis and death, generally within 2 to 5 years of diagnosis. So you might be sitting here thinking, well, you know, if we know that the upper motor neurons and the lower motor neurons are dying in this disease, why hasn't anyone actually stopped that from happening yet? That seems like a pretty good question, and it's one we ask a lot. And this is a best example to me of why we actually haven't fixed this problem yet. and that is the corticomotor system is incredibly complex. So this young man's corticomotor system is incredibly fine-tuned, and you can see and think about how many different processes need to go into doing this slam dunk effectively. You could think about his calves and his quadriceps, which would be very well defined, you can imagine, but you think about them firing and the need to jump and all the muscles that require for him to do this. So that needs the lower motor neurons to fire and do that. But when we think about this, and the other thing you need to think about is that where does this signal come from? It comes from the brain, and something in the brain is telling him how to jump, where to aim, and that sort of thing. And the other thing that makes the system so complex is that we don't only have go signals. And so upper and lower motor neurons are what we consider go signals. They're the things that initiate the firing of a muscle. However, we also have what we call stop signals, and other neurons cause these stop signals. And the stop signals allow this really fine tuning of the corticomotor system. I kind of think about not having stop signals is how my 5-year-old drives a radio control car, because he doesn't have very good skills yet. And so he's just always going straight, straight, bang, straight, straight, bang. Without the stop signals in our corticomotor system, we can't fine tune and we can't do these really elegant things. And so it's a really complex balance between go signals, stop signals, your brain, your spinal cord, and your muscles that allow all this to happen. And this is exactly the problem in motor neuron disease, is that we don't know what cells are dying when. We don't know exactly what's going on, and we haven't really got to the bottom of what cell types are impacted and when they're impacted. And so there's a lot of energy and a lot of research trying to work this out. And so there's actually what we call two different hypotheses that exist in the motor neuron disease field. And you might have heard of these if you've done some reading, and from our last session, by the sounds of it, everyone is very well read. I was incredibly impressed. And so these two hypotheses are important to understand, because if we can work out what is the first trigger of this disease, where does this where does the biology start, that might start to tell us where we need to treat. You know, should we be thinking about the brain and what's going on in the brain? Should we be thinking about the spinal cord? Should we be thinking about the muscle? Or should we be thinking about it all as one and it doesn't matter? These are really important questions, because what we need is effective therapies. So we call this the dying forward and the dying back hypothesis. And they're pretty self-explanatory when you think about dying back. Is there something going on in the muscle that's then causing all these neurons in the corticomotor system to stop working? The opposite could also be said to be true. And there's actually a lot of energy in Australia looking at the dying forward hypothesis. And this is sort of what I'm going to talk to you about today. We're very lucky. We've got some really brilliant neurologists in Australia that have really led the way in this understanding of what's going on in the motor cortex. You might have heard of Professor Matthew Kiernan, Professor Steve Bukic. They've done some really lovely research to show that, you know, changes in the brain is one of the earliest changes that could be happening in patients with ALS. So, you know, we kind of have what we believe now as researchers and clinicians is that there's some good evidence for both. And so, you know, all good hypotheses can only be can only be supported, they can't be proven. And so, but we're still trying to work out and understand what's going on. And, you know, and it could be that actually neither of these true. You know, it could be that all the cells are independently dying and there isn't one origin. But it's important to understand this and understand how to treat disease. So, unfortunately, we still do have quite a few unknowns in motor neuron disease. And as I've touched upon, we don't really know if the disease begins in the brain or spinal cord. Presentation of motor neuron disease can be really vastly different for different patients. And this makes researchers' jobs a little bit difficult in the sense that it's hard to know, you know, not the disease isn't the same for everyone. And so understanding how the disease starts, how it progresses is still something that's really complex and trying to tease out. Now, we believe that motor neuron disease is really multifactorial. And I think this is a great example of, you know, and I'm very biased, but MND researchers are really nice. So in Alzheimer's disease, there's basically two camps. You're either pro that beta amyloid hypothesis or you're anti it. And there's still quite a lot of, I guess, a disagreement about what could be going on in Alzheimer's disease. In motor neuron disease, we kind of all agree that actually there's probably not one right hypothesis. Probably what's happening is there's a lot of different disease processes happening, and they're kind of converging on this degeneration of the motor neurons. And so, you know, working together, understanding everyone's kind of expertise and disease mechanisms will be really critical. And, you know, starting to think, and the field is really starting to move forward to that concept of, you know, how do we treat these things together rather than trying to find a magic bullet just for one disease mechanism. The other thing that is really interesting about motor neuron disease is 90 to 95% of cases still are considered seemingly sporadic. We are getting better and better at understanding the genetics. And so, but it still seems that, you know, there are no known genetic causes for a majority of the cases. So this tells us we have to think about disease mechanism a little bit more when you think about treating. And unfortunately, there's no effective cure. We do have some treatments, but, you know, none of them are giving us that convincing delay in disease symptoms and that stopping in progression that we really want to see. So the other really interesting thing about motor neuron disease, and the thing I spend a lot of time thinking about, is there's this protein called TDP43, and TDP43 actually is present in a lot of cases when you look at cases post-mortem. And so this is just really striking. It really suggests that this protein is really important in motor neuron disease. And unfortunately, though, as a motor neuron disease researcher, our life is never easy. So not only is the corticomotor system incredibly complex, TDP43 is also incredibly complex. It's known as a DNA RNA binding protein, and at last count it has thousands and thousands of binding partners. And so this means that it can be involved in a vast array of different things. And so as a community, we haven't really had a lot of luck in understanding what this protein does in disease by trying to work out what it binds to. We know it's involved in a lot of different things, and also it's involved in neuronal health and communication is what it's really critical for. But this is obviously a lot of different things. So what we see happens in motor neuron disease is that we see that this protein moves. So if you can see these little red dots here, this represents TDP43 normally in a motor neuron. And normally it lives in the nucleus. This is where it does a lot of its action, a lot of its DNA binding action. But it also can shuttle between the cytoplasm, which is this part of the cell body, and the nucleus. And so it normally shuttles between here. It gets made in the cytoplasm, goes back into the nucleus, has a little bit out here from time to time. And this is its normal function. However, what happens over time in motor neuron disease is that it starts to really build up in this cytoplasm, and it stops going as much as it should into the nucleus. And so not only is this protein incredibly complex in the sense that it can do a lot of different things, it's also complex in disease because it starts building up in the cytoplasm where it shouldn't be. So it's telling us that it's in a place that shouldn't be, but it's also not going into the nucleus the way it should be. So we're losing it from one place and having too much of it in another place. So there's still a lot of work trying to tease out, is both of these things a problem? Is one of these things a problem? And what's the most important thing? And so this is basically this is where the big questions are, and this is where my research is at. And, you know, and as a community, something we're really trying to understand. I really like this picture, even though it's incredibly confusing. But I thought I'd just show you there to kind of get a hint of why we need preclinical research and why we do preclinical research. Is that it's very hard to understand what's going on with a protein such as TDP43, which does a lot of things, plays a lot of roles, but also can be in different compartments. So it can be in the nucleus, or it can be out in the cytoplasm. So when you sort of, if you look at it at disease end stage, you know, what you might see something like this. So normally TDP43, which is in brown, is just normally in the nucleus. But in end stage, you see there's some in the cytoplasm. There's some that's aggregated in the nucleus or the cytoplasm in different cells, and other cells also are involved in this. So it's really complex and hard to tease out. So this is where we need to start asking questions in the preclinical space to try and really pinpoint what's going on. And that's what we can do. Now, I don't want you to feel like you need to understand the molecular biology here. I just wanted to kind of give you a feel of how preclinical research works and what we do. And so I really want to know, you know, what happens when you turn TDP43 on in the brain. If you turn it on in the brain, do you see changes going through the corticomotor system? This is important because we don't know if the brain, if TDP43 dysfunction in the brain can trigger motor neuron disease. And if we can show this and understand this, this might tell us what we need to be doing is treating TDP43 here. I do this by using a mouse model. We use a lot of different mice models to investigate motor neuron disease. Obviously, in animal research, and using animals in research is a privilege, and we only do it when we can't do a similar experiment in a dish or with tissue. And so because the corticomotor system is so complex, this is a situation where we do need it to be intact to really understand it. So basically what I can do is use some complicated genetics, use different ways to express this complicated genetics, and actually turn TDP43 on in certain neurons. And that's exactly what I do. And basically, again, I won't go through this, but I just want to share my love for the motor neuron with you. So this is a lower motor neuron, and this is what I look at in the microscope. Well, that's a bit of a lie. This is what my students look at the microscope and my postdocs. Now I spend a lot more time on my computer, unfortunately, but this is what I really love to do, is stare at lower motor neurons. And you can see this is you can see that they're really beautiful cells here. And by using this complicated system, I can turn it on only in the motor cortex, and you can see there's no expression in the lower motor neurons. So then we can map it out, and we think a lot about, well, what does this actually mean for disease? What does this mean for the patient? And so we set up our experiments so that we're replicating what is happening in the clinic as best possible in the mouse. And we think about, you know, what's the timeline and that sort of thing. Now, a mouse ages a lot more quicker than a human. And there's been a lot of good developmental studies that tell us how old a mouse is and the relative things. And so we can then start, okay, you know, if progression is 2 to 5 years, let's turn on the protein for about 30 days, which equals about 3 years. And again, this is just this beautiful expression in vitro. And we can ask really specific questions. And so I'll just show you a little bit of data quickly before I run out of time. But basically, what we found, which we're really excited about, is that if we turn TDP43 on in the brain, and it's only in upper motor neurons, we actually see a loss of lower motor neurons. So to us, that is supporting the die-forward hypothesis, that something going on in the brain can cause a problem in the spinal cord. But this is where the molecular biology gets a little bit complicated. But what we're really excited about is that not all lower motor neurons are equal. They don't all die at the same rate. And this, I'll skim through this so I can get, so I have a lot of time for questions. But basically, what we're seeing is that different types of neurons actually degenerate quicker if in the spinal cord compared to the brain. And we then looked at excitotoxic signals and tried to work out what's going on there. And what we actually find is that there's a real regional patterning of degeneration in the spinal cord. And this is important because this is the only way you can see this sort of thing is basically in the lab. Is that, you know, we would have missed this had we not turned on the protein and slowly watched it every day to see what is happening. And this really laborious watching of degeneration in the spinal cord is what has revealed these really exciting data for us. And so we have found the evidence, and we're currently trying to publish this. And we talked a lot about reviewers this morning, and I hope we have some nice reviewers at the moment. But we've discovered that pathogenic changes in the brain can spread through the corticomotor system in a die-forward-like manner. But however, there's this differential vulnerability of different parts of the spinal cord. Now, when you think about patients, this is starting to feel very interesting, because we know that every person's different, and how the disease is affected is very different for different people. And if you start thinking about, it could be due to different differential vulnerability, different parts of your spinal cord might be impacted a little bit more than other parts. It'll really depend on where the disease has started and where it's spreading. And so this could really help us understand how to treat the disease, but also what the other questions we really want to know is, what's this molecular signature that makes one cell more vulnerable than the other? Because if we could really tease that out, then we might be able to know, you know, we might be able to take the characteristics of the cells that aren't so vulnerable and use those to help the vulnerable ones. And so I guess this leads us to a really big question about where do we move on from here? And so this is a timeline I talked to you about in a mouse, and we think this is kind of around 3 to 5 years in a human. But what we find that's really interesting is that when you think about riluzole, you know, riluzole treats a really important disease mechanism in motor neuron disease, but its effectiveness doesn't always, it's not as effective as we would like, and the effectiveness wanes. And now there's a lot of hypotheses about why this effectiveness wanes. But what our research has suggested that it might actually be to do with not only the fact that we're probably not getting enough in there, or that the cells might be changing their response to the actual drug, but we think that actually maybe the disease is changing over time. And this is really important, because if you think about the upper and the lower motor neurons, we see this lower motor neuron loss in a really selective way. And so it's making us think upper motor neurons and lower motor neurons actually might be a little bit different to each other, and they might have a different vulnerability. And so perhaps what we might need to do is start treating the lower motor neurons or the upper motor neurons as different cell types. And we really want to know, you know, should we be treating these guys first, or should we be treating these guys first? And should we be trying this really a precision sort of targeted therapy, where we go, okay, the lower motor neurons need this type of therapy and the upper motor neurons need this type of therapy. And this is where my research is at the moment, trying to think about novel cutting edge ways to, rather than provide a drug that treats the whole corticomotor system as one, but actually provide different parts of the corticomotor system with different treatments for what they really need. And so with that, I just obviously have to thank all the people that are doing the research. Trace and I are the ones that lead the research, which means we don't actually do a lot of the fun stuff anymore. And without our funding, which MND Australia is a critical part of my funding, and it's certainly kept my research lab alive for a long time. And so, you know, we're really passionate about this disease, and I'm happy to answer any questions that anyone has. Thank you. And I'll stop sharing.
Thank you, Kathy. Awesome overview, and yeah, very well. You responded very well to the difficult title I gave you. Yeah, that's fantastic. So I just saying, so you think going forward, we'll probably have a kind of a personalized medicine type approach, certainly in the near future to MND, and sort of almost look at every patient and go, it looks like these systems are disrupted, and perhaps you'd have a treatment for your particular kind of sort of flavor almost. Is that how you?
Yeah, definitely. That's exactly my dream. And I think we need to be working with our clinicians, which we are. So we need to be able to, we need the readout at the same time as we need the drug. But that's what I think, if you, it could be with TMS, but we need to think about how we can target the lower motor neurons as well. But if you got a picture of what was going on in this specific patient, then you could say, okay, well, for their brain, this is what we need. For their spinal cord, this is what we need. And then working out ways to actually deliver that specifically to the brain and spinal cord could be really effective. Because it could be actually that something that works on the brain is actually going to be bad for the spinal cord and vice versa. And so this is, why we think that, some drugs, a lot of drugs kind of are failing that leap between the lab and the clinic, because disease mechanisms may still be important, but they might not be important for all motor neurons. And so how do we get it to the motor neurons that need it the most?
Brilliant. Peter Clark.
Yes.
Question?
Do we know why some people like me, it's all upper motor neuron loss, and most ALS is both? Like, why didn't I get lower?
That's a really good question
as well.
Yeah, and so these are kind of questions that we're really still trying to understand. And so different motor neurons do have different involvement of upper and lower motor neurons, but as to why, it's so hard to understand. And it's it's understanding disease progression is, it's what we're sort of really trying to sort of get to the bottom of. And I think this is sort of, when I mean, it was a while ago now, but when I first started working in neuroscience, I said to my boss, I want to look at, it was in trauma actually, but I said, I want to look at different neuron types. And he said, no, nobody cares what kind of neuron it is. All neurons are the same. And actually that's really changed now. We know that's not the truth. We know that neurons are very different. And so it's a really new field, and trying to understand why some neurons succumb and some don't, it's perplexing, but hopefully it might lead us to understand disease better, but also understand, if we can really nut out why some neurons don't succumb, maybe we can find, some sort of answers there as well.
Matt, you have a question?
Hi, Catherine, and thank you for the presentation. I took a lot away from it. I have two questions. The first one, your research supports the die-forward hypothesis. Does that then exclude a die-backward hypothesis? And the second part is, how can someone like myself aid to contribute to your research?
So the first question, thank you. They're both really good questions. The first question, it's a very good question, and it's one that I didn't touch on, but I had a lab meeting the other day, and I told everyone my vision for the next 10 years, and they were like, calm down, Kathy. But I think, this has been really perplexing for the field for quite a long time. We have really strong evidence for the die-back hypothesis, and now we're starting to get strong evidence for die-forward hypothesis. How can we have evidence for both things that are kind of counterintuitive? And there's another hypothesis that doesn't have a lot of evidence to it, but I'm kind of starting to think about a lot, is the independent hypothesis, that upper and lower motor neurons actually are differentially vulnerable. And this is what I think I'm seeing in my research, is that this is why you can have so much evidence for two counteractive hypotheses, is that perhaps both of them are a little bit wrong and a little bit right at the same time, in the sense that that the neurons are actually dying at different rates. And so they're actually, even though that they're spreading disease, and even though they're communicating with each other and spreading this dysfunction through the corticomotor system, different neurons respond differently to this dysfunction. And so it's actually thinking about them as independent, and that the upper and the lower motor neurons are degenerating independently. And so that would be, this is, I guess, all I can say, this is a best guess, so that's not a perfect answer, but that my theory is why we have so much lovely evidence for both is that actually there's a third hypothesis in play, that they're actually independent. And the second question, so I think that's probably going to be more elegantly answered in the next session, where we start talking about advocacy, and and we as MND researchers, we really strongly believe in the involvement of patients and people that have a lived experience. It's critical. It's critical for forming our research questions, keeping our research questions on track. And it's actually amazing what you can learn from patients about the disease that kind of really can inform your research. And so I think MND Australia do a great job. So we have there's research forums, and and forums like this, just being involved and telling us things about the disease is really important. And so I'm kind of always really appreciative of people's time, but I think we'll probably get more into that in the next session as well.
And on a sort of more personal basis, I guess you can then sign up for studies which are applicable. I mean, certainly make sure you're in the patient registry, MINDOS, so your data is hopefully being harvested into that. Hopefully I'm not stepping on your toes there, Karen, for the next talk. And Rob Henderson, who is, I think Rob might be your clinician. He's certainly involved in both trials, but also studies as well through UQ, looking at measuring different areas in patients to see how they're progressing and how they can relate that back. So as long as you're sort of able and willing, then if you see a trial that you think or a study you want to be involved in, jump in there, because the more data we have, the more we can test all these hypotheses. That's really critical. It's just accumulating information from as many people with as many different forms of the disease as we can, so you can then just build that data set up. Unfortunately, it's a numbers game. It really comes down to it, I think. If Kathy, you had anything else to comment about that? I mean, clinical samples, basically, and data.
Yeah, I am involved in quite a lot of the research programs with Rob Henderson already. I'm just not sure how far the reach goes. I'll discuss it with him as well.
Great.
Jojo, you had a question as well?
Yeah, it's for Kathy. I just want to make sure that I heard this correctly. Did you say that the protein degenerates quicker in the spinal cord than it does the brain? And if so, can you explain the pathology of why that is?
Great question. I said that the neurons degenerate quickly. So when we turn on that protein, the lower motor neurons are lost at a quicker rate than the upper motor neurons. Why? I just recruited a new PhD student, and that is exactly her project, is to kind of tease out what is the difference. And so we have to make sure, it could be the signals they're getting, it could be the way the protein is impacting the lower motor neurons. It could be that upper motor neurons are a really big group of cells, whereas in the lower motor neurons, they're very, very discretely organized already. And so it could be that we're just finding the ones that are more vulnerable first. So it's basically, I've just explained the 3 aims of her thesis. And so it's something that's really novel that's coming out of our group. And so we're now trying to understand why that is. And hopefully, you know, it could be that there's subsets in the spinal cord. So the fast fatiguing that we see is more vulnerable in motor neuron disease. It could be that they're got the ones that are going first. And so they're kind of really big research questions for us.
Thank you.
Thank you. All right, in the interest of time, that was a fantastic discussion. We'll now move along with Karen. So while Kathy is working out what's going on with all the different motor neurons, Karen's going to talk to us about all the research that's also going on with making sure that we're providing the best possible care to those who are living with MND right now, and how we can keep advancing that care to make sure it keeps improving. So Karen, please take it away.
Can you all see that that slide?
Yep, your screen's good and your sound's good as well, Karen. You're
Yeah,
good.
this is the trouble when you're looking at 2 screens. I'm not quite sure which.
Yep.
So thank you. All right, so I'm just thank you so much for the opportunity to present today and provide an overview of some of the healthcare research being done currently in Australia. So this is just a bit of a snapshot. And I'm not involved in all of this sort of research, so I'm just giving you some information as to what is happening. Some I am involved in currently, and I'll explain a little bit more about those projects. But I'm a postdoc research fellow with the Macquarie University at the Australian Institute of Health Innovation, and I'm also a physiotherapist, and I've worked in the UK and Australia with people living with motor neuron disease. I have a sort of particular clinical focus in working with people with neurological conditions. So let me just get into it there. So you've probably seen this quote going around. It's quite a quote that I think is very apt in the healthcare space, is until there is a cure, which is what Kathy was just talking about, all her amazing research and all the other researchers that have talked about how we can work towards getting a cure. There is also care, which is very important. And we're looking at people and family-centered care according to specific needs and priorities. And the little diagram at the side is that we all know that everybody is individuals and so have different priorities and needs. And it is important to consider living well with motor neuron disease throughout all the care needs. So I want you just to have a little look at this diagram. This isn't specifically anything to do with MND, but it just gives you a bit of an insight. So what I want you to have a look at is this very, very busy picture that was drawn by a young girl that I know. And I just wanted to, I feel like it's a really great way of looking at the busyness, and this is what healthcare is about. There is so much going on. You have to navigate so many systems, and it's actually, we need to sort of gain understanding and to know what needs to change. So I just wondered if you can all see the owl in the picture. So just have a little look. And sometimes things aren't always obvious. We have to sort of actually delve deeper to find out what's really happening. Did you all find the owl? But there it is, and there's so much detail that you could spend a lot of time observing and looking and listening to experiences and finding out more about what is happening in the healthcare space. So I feel that it's really important just to sort of go over the different ways that healthcare and social care is funded within Australia, and I'm sure you're all quite aware of all of this, especially the National Disability Insurance Scheme, the NDIS, if we're going to abbreviate it. And that's for people under 65 years, and that becomes under the disability sector. And then we've got the My Age Care, which is the aged care sector, which is for 65 years and over. There's the not-for-profit, the public health system, there's various private health insurances. So there is really important to understand that this has created some complexity, or quite a lot of complexities, particularly conducting research. So a background on what do we know? What is considered quality services in healthcare? And there's been discussions about multidisciplinary clinics are optimal approaches, and they can actually help to improve quality of life and care experiences. And these should be person-centered and integrated across the public health system into the primary care, community care. But as I've already mentioned, we've got these current funding models of Australian care that actually bases the funding on age rather than actual disability. So which can create barriers to equitable care and timely and efficient and connected care for people with MND. So this is a quote about the National Disability Insurance Scheme. It has been developed as a plane being built during a flight. So it is something that we need to sort of consider, and that it is under review again at the moment. And also the locality of specialised services, most are within the cities around Australia, which can be a barrier for people living in rural, regional and remote areas. But this has, is being addressed more and more in the use of telehealth, which does seem to be a growing area of research in this area. And we have more research in that. I'll go over that in a minute. So this is for many people that live with motor neuron disease, and if anybody has seen some of my presentations before, this is something I just wanted to highlight, that these services should be similar for all people living with MND, but are not always connected very well. And it sort of, the care can be a little bit disjointed depending on where you live and what funding sources that you have. And sometimes there's a lack of coordination and communication across the different disciplines. But this is where and I've highlighted, it's got a bit of a glow, the specialised MND clinics can give, be more of an opportunity for that partnership, develop more communication and work together with the person with motor neuron disease and their family members. So I just wanted to go over what is integrated care, because that is something that's often talked about. So it overcomes fragmentation of care delivery, so that it's coordinated and according to need. It's people-centered and population-orientated, and it improves quality of care and cost effectiveness. And it takes a person-centered focus to services and the way that they're innovated, and individual and family caregivers and the wider community that they belong. So for people that might be aware, this is the vision for healthy Australians. This was done by the Australian Healthcare and Hospitals Association Blueprint. And I just wanted to focus on, the health services and truly integrated and connected. So they're talking about efficient models of care, the health workforce, hospitals are effectively utilised, and participation of individuals. So this is a whole partnership. So we're all aiming for what has been described as the quadruple aim in healthcare research. So it's better outcomes, improved provider experience, improved patient experience, and lower costs, which is the aim of most of the healthcare research. So this is a bit of a snapshot of healthcare research currently being conducted across Australia. So as has already been mentioned several times, is MINDAUS. And just a reminder again that just Gethin has just reminded everybody about the patient registry, which does enable, you know, yourself, family carers and service providers to track their disease progression and, you know, changing support and needs. And also it's a good way of getting consumer input into service design and evaluation. And the goal is to improve care and coordination and develop a national framework, which is what we've just been talking about, which is a government, a whole national government initiative is around integrating care and improving care and coordination. So the main focus within MINDAUS with regards to policy is improving health literacy in MND, care integration, and again, models of care that are equitable across the whole group, the population group living with MND, improving workforce capability, which is another area, addressing carer needs, and also the benefits of MND patients remaining at home. And I've just put the website in there for anybody that might be interested in looking it up. So currently I'm involved in a rapid review, which is exploring best practices of integrated care models in MND, and this is around the international focus. So it's not just an Australian focus. And so these are some of the papers that have been highlighted in this review. So I'll just give you, so the aim of the review is to identify models, approaches to integrated care, and sort of compare and review the models, components of models, and how they contribute to more effective and efficient person-centered care. So we started off, and this is like a prisma diagram that we use in reviews to sort of outline the process. So we had 1,534 references to start with, and then the duplicates. And then we've done a title and abstract review, and we're currently finishing off a full data extraction of 57 studies. So what does the literature say about integrated models of care? So there's basically no standard international standardized approaches in design and implementation. There's consistency in multidisciplinary workforce and the degree of integration and partnership between hospitals and the community. So we're all quite aware of some of the benefits of these coordinated and integrated MND clinics, improved health outcomes, improved resource use, improved survival in MND with more timely access to non-invasive ventilation, for example, and gastrostomy, inclusion of palliative care services, communication across organizations, and the link with community-based NIV, which is non-invasive ventilation, and mechanical ventilation teams. So some of the considerations that was the distance to specialized MND clinics and the time taken in those clinics. The multidisciplinary teams need to consider the stage and duration of MND and the expectation, needs and priorities. And education opportunities need to be considered to upskill health and social care providers as well. So currently there is a study being done in a collaborative study, which is exploring access to motor neuron disease healthcare in New South Wales. and the potential use of telehealth for patients in remote, rural and regional areas. So these are the 4 objectives. So the need to confirm the need for improved access to coordinated multidisciplinary specialist care, particularly focusing on those in remote, rural and regional New South Wales, and to report on the areas of greatest clinical needs, the challenges and barriers faced by healthcare professionals, and they're the ones that are not working within the clinics and maybe are not have not been involved much with people living with MND. We have sort of skills shortages across the rural and remote areas, so any support and education is really important. And also they're looking at the specialist MND virtual health model and looking at the experiences of people that have used this service, and also the willingness to engage in this form of clinic. This is one that I am involved in, is evaluating the role and impact of a regional motor neuron disease multidisciplinary clinic. Again, this is a collaborative study, and both these studies have been funded for about a year, and it's small amounts of money that we've been able to obtain through through different grants. And then they examine the barriers and enablers to implementation and sustainability of running this regional clinic, and the information to inform and design and help others if it's felt that it's a positive development on other areas developing this model. And this is where it is, it's an hour and a half to 2 hours north of Sydney in the Central Coast. So currently we've held 9 clinics, and we are finding that without any advertising of the clinic, that we've got increasing numbers of people wanting to engage with the clinic, and it's run every 4 months, and it is run out of a private clinic at the moment. So some of the preliminary findings on the implementation barriers and enablers is that we have been able to access a private clinic space. We have got strong relationships and partnerships with people, allied health and medical teams working in MND, and we seem to have all been in this area for a long time. So there is quite a wealth of experience in that area. But everybody that has attends the clinic is not attending as a public health employee. It is in private private time. And we have administration staff. We've got good connection with people living with MND and the families, and access to technology, and there is a research trial being conducted at this clinic too. And we're very lucky. We have very strong connections with MND New South Wales advisors and coordinators of support in our area. So one of the barriers is lack of engagement from public health, the local hospital, and the healthcare professionals within that LHD are not able to actually attend the clinic, so they attend via telehealth. It impacted the sector and funding silos. It's currently not funded, the clinic, so that's going to affect sustainability, and limited capacity to run the clinic more frequently to meet the demand. So we've got other areas of research, communication and decision making. So the impact of communication on healthcare is an area that has been, so Camille Painter and Susan Mathers have done quite a bit of research in this area. So the communication impairment support and accommodation. Otherwise, healthcare involvement can be compromised. So education and information may prompt clinicians to consider the best way to conduct clinical consultations to accommodate patients' abilities, ensure they ask their preferences. So this was a study that's come out fairly recently, last year. The use of, and the next one is similar authors, and the results highlight the potential benefits of gaining an early understanding of and accommodating communication needs, personal preferences, and emotional readiness for information for people living with MND and their carers. And I think that's certainly something that comes out as a healthcare professional. We know that for some people, you know, the timing of the information and how the information is presented is very important. This is another is the decision-making tool, and Anne Hogden has done a lot of work in this area. The model provides a framework for patients, carers, and healthcare professionals to enact patient-centered care, incorporating their healthcare and psychosocial needs. And this model has gained international attention, and it aims to optimize timely decision-making by prompting early and recurrent discussions of symptom markers and time points for action within each stage of the cycle. And this is regarding the same sort of model, how healthcare organizations can better facilitate gastrostomy decision-making to meet the needs of people living with MND. And this was so they're suggesting that there needs to be more research in this area and to better inform clinical guidelines and minimize the impact of delayed gastrostomy insertion. Another area that there is a group of people that I've sort of know of and been working with them in the MND are the collective that Gethin had already mentioned, where we sort of under the Clinical Care Collective. And so this is a group of researchers that are looking at non-invasive ventilation uptake. So they've found in Australia that only 19% of Australians with MND ever try non-invasive ventilation. And it is currently the most effective therapy for extending life in motor neuron disease. And I know that it sort of ranges between 5 to 13 months, seems to be what the research is saying. So there's different research going on in this space to try and encourage and you know, more uptake in non-invasive ventilation. So we've got Dr. Marnie Greco, who is currently doing some research in improving health outcomes in motor neuron disease by, again, the uptake of NIV. There's looking at polysomnographic titration of NIV ventilation. There's non-invasive ventilation at home, which is looking at comparison whether it's introduced in a clinic or in the home. And the researchers found that there is really no difference in the pilot study with regards to quality of life or carer burden, and with regards to the NIV being introduced in the home or the clinic. And then there's the Repair MND, which is looking at artificial intelligence projects aimed to develop software that can support, again, the optimization of NIV. So this is the project. So within the initial project that I mentioned, there are several aims, and one of them is actually to get an accurate estimate of the current rate of NIV uptake. And that again is through a survey, understand the barriers. So this again was looking more deeply, delving deeper into finding out why this is the situation. And then looking at ways to overcome those barriers and implement that, whatever the tool or the method to sort of try and improve the effectiveness of this strategy. So a focus on palliative care and caregivers is another area that has got large prominence in Australia. And all these research areas that I've sort of mentioning are all being highlighted in the integrated care review that that I mentioned at the start. And it is all these areas that are being picked up in Australia that have been that are necessary to sort of develop more integrated care. So we've got so Samar one, I'm not quite sure how to say her surname, so I'm sorry if I pronounced that wrong. She's involved in all of these researches in Western Australia and looking at compassionate communities and the connectors of those communities is becoming very important, realizing that, you know, that recognizes death and dying as everyone's business. And that they should be partnering in the long and complex task of providing care, quality of care at end of life. And looking at grief and depression in a study, and it highlighted that there is 63% required bereavement support over and above that what was required in family and social networks. And significant prolonged grief disorder was predictors of that were poor family functioning, and also that the fact that the length of the disease journey and the age of the person when they passed away and the period of time of caring. And then also we're looking at bringing motor neuron disease associations, how they can actually support and work together in improving the time of bereavement before and after. So healthcare research needs you as partners to make these changes. So people involved in healthcare research include the health and social care providers, healthcare managers, people living with MND, family members, including children. We need to include the children. It's very important that we take a family centered approach because they're involved in, and often they're a neglected group. They're young carers, and it's really important that we highlight this population, and also the community groups. And I mean, there are more people, and the range of methodology used in healthcare research, and that's been mentioned, interviews, focus groups, surveys, random controlled trials, observations, studies, and pilot intervention studies, reviews, medical reviews. So there's a real range of ways that people can engage in this type of research. So just finally, I hope I haven't gone over, working together to improve care experience, living with MND. It takes a whole team to do this, and thank you.
Thank you, Karen. Great overview. It's just showing I think the importance of trying to bring. There's a lot more research, I think, in the care space than sometimes we realise, and I think it really highlights the importance of trying to bring everyone together to make sure that we're kind of talking to each other and we realise what's happening, and bringing people together right across the full spectrum. Just one quick question. So do you think then by in sort of more remote areas that having like a really, really good coordinator can give you almost as effective treatment as a multidisciplinary clinic by just coordinating all the different services, all the different allied health and neurologists,
Mm.
Can that overcome perhaps that geographical barrier and really mean that you do get that almost the equivalent of a multidisciplinary clinic by just having someone who's really good at organizing stuff?
Yeah. I certainly there in our review that we've just done, there's an interesting paper, and it is about a telehealth trained nurse who actually does the whole multidisciplinary assessment and then goes back to the team. They review it, and then they have another telehealth session with various team members and discuss the plan of action, and what to address any symptomology, or all that sort of thing. So that's a really interesting area, and care coordination certainly is something that we know works, but there still is a lot of research out there trying to prove that it works. But it is an area that is certainly would be very valuable and help people navigate all the complexity of healthcare.
And does the Australian system provide an avenue to fund that, the healthcare system, or does it really have to come through the state associations or other organisations such as ourselves?
So under the NDIS, you can have a coordinator of support, and they can be under the Motor Neuron Disease Association, but some people choose a care coordinator that's not attached to the MND Association. And that can be, I've worked with people in that situation, and that can be quite challenging because they have, they might not have the insights into the needs of people living with MND and the family. Under My Aged Care, I think the MND advisor tends to take on that role, and they are absolutely valuable in helping people to navigate the complexity of the care needs, and also the challenges, is particularly with aged care, with the different packages that you can get, the length of time that you have to wait for those packages, and also linking people into the public health system to get allied health, and I know they've been real big promoters of our clinic that we've been that we've implemented. They've been really trying, because then we are able to coordinate people's care and actually get them into, say, palliative care or respiratory care, or because some people aren't actually linked in with those services.
Thank you, Karen. We've got time for a couple of quick questions then from Michael, then Jenny.
We've just been through that sort of exercise with My Aged Care over the last 3.5 years, and everything you said is true, but on steroids.
Yes, yeah. I think the clinic, attending this clinic, has really highlighted to me the huge inequities in care and equipment provision for people under the aged care sector. It has been quite challenging seeing how much the difference.
Too little, too late.
Jenny?
Thanks, Catherine. Karen, so much of what you said was so heartening. There are a couple of things I wanted just to touch on, just in view of my own experience at the very beginning of this. A lot of what you were talking about was communication information. In terms of navigating this from the outset, I live in a metropolitan area, obviously, so our resources and the access to information services should be good. Navigating it has been really, really problematic and complex. I do worry for anybody who might not have access to that, or who might not be as readily able to understand the situation, where to go, where to look for information, etc. I think one of the things that we would have found enormously helpful would have been a medical pathway. So a guide to understand exactly how this all should come together from the outset. I've had to try and put that and piece that together myself. Having a GP who I'm the first person that she's encountered, and so she didn't know, and she's been on this learning journey with us. Being sent away by a neurologist initially with no information, and being told by myself. What we found, because I've been, as mentioned before, I've been proactive about this. I engaged with M&D Australia and M&D New South Wales. We had an invaluable conversation with the M&D advisor, who helped connect us with some resources around how to navigate some really critical decisions and conversations that we knew we needed to have with our children and with our children's schools. We've just done that in the past week. But all of that could have been so much easier had we had a pathway and a guide. So I think there are some really important communication resources that should be put together for patients and should be rolled out to GPs, to neurologists. I think there's a lot that can be done in that area to improve this process. So the work that you're doing and raising the profile of these sorts of things, I think is critical, and particularly the equity in rural regional areas. So thank you.
Thank you. And thank you, Jenny, for bringing that up, because we do know that GPs, you know, they might just see one person with MND, you know, that might be the most they may see. And it is, again, as you said, it's been a learning journey for them as well. And we are, you know, we are very aware that about clinical guidelines, actually having that, you know, understanding of what, you know, what is the processes, where do people go? But it is really complex. And I totally agree with you. There needs to be much more work done on that area. So thank you.
Christine, last question.
I guess, sorry, I found this quite upsetting. I just wanted to pass 2 comments. One of them, my mum experienced MND in New Zealand, and it was a regional area, and we were not open to any of the care that you discussed. So that's quite confronting. And all of my family have turned their back on MND. They don't want to be speaking for it. And they're surprised that I am. But the other comment I wanted to pass is having an uncle and a mum that had it, we don't think it's genetic because nobody else in my family has it. But as a Kiwi living in Australia under the age of 65, I do not access the NDIS, and there are 500,000 Kiwis in the same boat. So this is also alarming to watch this, to know that I have a barrier to that, even living in Brisbane in a major city. So that's confronting too.
Yeah, thank you. And I mean, I think that's highlights a lot. There, you know, there's been a lot of different population groups that are challenged by this, and I'm sorry, as I said, these are the services that people should, you know, be entitled to, and unfortunately not everyone has access to those, so I'm sorry to hear that.
Thank you, Christine. So this actually probably takes us very appropriately into the next session now, which is about advocacy, and obviously there's quite a few topics that were raised there that probably fall under the umbrella of advocacy. So I'll just pass over to David Ali, the CEO of M&D Australia, who's going to moderate the discussions around advocacy. All yours, David.
All right, thank you, Gethin, and it's good to be here. Unfortunately, I've not been able to tune in today nor yesterday because I'm at the National Rare Diseases Summit here in Sydney, which is a combination of clinicians, people with rare diseases, medical doctors, researchers, people like me. Thinking about the National Strategic Action Plan for Rare Diseases, of which MND is classified as a rare disease, and thinking about it in context of awareness and education. And so, Jenny, to your point, it's not only awareness and education for GPs and allied health professionals and others, but it is also kids, and doing education in schools about rare diseases. The second aspect is around care and support, and the third is around research. So it's been absolutely fascinating. But recognising there are 7,000 rare diseases, it's how does one actually get their voice heard? And that's a real challenge when you're working with time-pressed people, like professional organisations, politicians, senior bureaucrats, and so on. So that's where advocacy comes in, and we've got two really great speakers, one being Carol Burks, who's the ex-CEO of MND Australia, so she's my predecessor, and she's probably going to talk more about the systemic focus around MND advocacy in Australia. And then Peter Chambers, in terms of what can you do as a lived experience advocate. So Peter will take, I think, that more personal view. So I think with the combination of systemic and personal, it leads to hopefully the same outcomes. So I'll hand over to you, Carol.
Thank you, David. Can you hear me okay?
Good. Yes.
Good. Okay, I'll just share my screen now, hopefully. Share. And okay, have you got the presentation up there now?
It's getting there. We can see.
We've got the slide view, not the you need to, if you click the little presentation button, I think you'll get the proper full.
Got it now?
Perfect. That's it.
Yep, great, thank you. Hard to see, as Karen said, when it's across two screens and you don't get it on the screen you expect to. Okay, well thanks, David, and hello to everybody. Thank you so much to the organisers for hosting this really important event here in Australia, and inviting me to talk today about advocacy. So as David mentioned, I was the CEO up until last year, from 2006, of MND Australia. Prior to that, I was the manager of support services at New South Wales, and I've also spent some time as the chair of the International Alliance of ALS MND Association. So I've sort of worked across the MND space, both at a state, national and international level. And prior to all of that, I was a registered nurse, so I've been in health for quite some time. I'm going to talk to you about advocacy, but I thought first of all, I might help set the scene by providing just a brief history. So the development of MND associations in Australia. So in 1981, the International Year for Disabled Persons, the first two MND groups were formed, one in Sydney, MND New South Wales, and one in Victoria, in Melbourne. Over the following decade, MND associations were formed in all states of Australia and the ACT, and the MND Research Institute of Australia was also established. In November 1991, MND organisations from across Australia met to consider the need for a national body, and it was agreed that the MND Association of Australia should be formed to undertake work to assist the state associations to provide equitable services and support. You've still got the screen there, have you? The presentation?
Yep, certainly do.
Just had a bit of a fiddle with where everything was sitting because I was looking in the wrong place. Thank you. Yeah, so November 1991, MND organisations from across Australia met to consider the need for a national body. And it was agreed that the MND Association of Australia should be formed to undertake work to assist the state associations to provide equitable services and to establish national advocacy activities. So from 1994 to 2006, national advocacy activities included submissions to inquiries related to voluntary assisted dying, superannuation, and a review of veterans' entitlements. MNDAA played a key role in working with pharmaceutical companies to mobilise community and government support nationally for the registration of Rilutech in Australia and its availability in the PBS. Then in 2005, a comprehensive external review was undertaken, which informed the development of a new ambitious MND Australia strategic plan with an increased focus on gaining recognition as the peak body in Australia. And in 2006, the first, what was known then as National Executive Director, then CEO, was employed on a part-time basis. And that's when I joined the organisation. So a little bit about advocacy. Advocacy is described as the deliberate process of influencing those who make policy decisions. There are three main types of advocacy, including self or individual advocacy, collaborative advocacy, and systemic advocacy. So it really does require a multi-pronged and coordinated approach at both at a local, state, and federal level, and also internationally. Most importantly, the involvement of people living with MND is crucial to engagement with decision makers and in effecting change. And I think Rick's talk yesterday certainly highlighted the power of research advocates already in influencing decision makers in the States and Canada. Sorry, it's not moving on. There we go. So why is advocacy a focus? Well, we've already outlined that national advocacy was identified as a major role from MND Australia from the outset. And in 2006, services to support people living with MND were a confusing mix of state and Commonwealth funded services. So even more confusing than they are today, that we've already highlighted how confusing it is at the moment. And there was also a very high level of unmet need. So despite evidence to confirm the benefits of multidisciplinary team approach to care, access to clinics was limited nationally. Access to services and support from MND associations also varied significantly at that time. Since then, MND Australia's close engagement with the MND community through its members, the state associations, has confirmed the ongoing importance of national advocacy to improve the lives of people living with MND. People living with MND need a rapid response and access to a range of services and support to address their complex and changing needs, no matter age, postcode, or funding stream, to maintain independence, quality of life, and the health and well-being of the carer. So over the last 15 years, advocacy efforts have focused on the need for a rapid response and equitable access to health and disability aged care services. Advocating for increased funding for research has also been a focus from the outset to support our shared vision of a world free of MND. As funding has grown and research has advanced, advocacy related to access to genetic services, clinical trials, assistive technology and therapies proven to slow disease progression has become increasingly important. Care with people for MND, as we've highlighted already, and also highlighted so well by Karen in her presentation, crosses a number of government sectors, including health, disability and aged care. And additionally, healthcare encompasses access to timely expert diagnosis, primary healthcare, multidisciplinary clinics, assistive technology, allied health, nursing and palliative care, and crosses both state and federal funding silos. This all makes MND advocacy particularly challenging, as having just one ask would be a lot simpler. With this in mind, we set about gathering evidence to build a case for change. Our strong national member network of state-based MND associations helps MND Australia to understand current issues related to access to needs-based care and the impacts of policy on people living with MND, their carers and families, and also the health professionals and service providers involved in their care. This ongoing engagement, along with state, national and international reviews, needs assessments and research, guides the development of key advocacy asks and builds the case for change. Developing networks and strategic relationships is also key to providing evidence to develop and prepare the case for change, as is understanding the policies of the government of the day. And with 6 elections and many ministry reshuffles since 2006, this has certainly added to the challenging nature of MND advocacy in Australia. As mentioned, an essential first step in preparing or presenting a case for policy change is to gather evidence. In 2015, MND Australia commissioned the Deloitte report on the economic impact of MND. This report has been vital in preparing and presenting our case for change. Following the publication and launch of the report at Parliament House in February 2016, an MND action framework was developed to outline the commitments we were seeking from federal politicians. The MND action framework was first shared with decision makers ahead of the 2016 federal election. This action framework has since acted as a guide to inform advocacy plans and community campaigns related to disability, aged care, assistive technology, genetic services, coordinated multidisciplinary and palliative care, and research. To best build on the evidence, advocacy campaigns must be multi-pronged, consistent, and sustained. Therefore, using the MND action framework as a guide, an advocacy plan was developed to include a range of strategies to effect change, as outlined on the slide and in more detail in the following slides. So, meetings with decision makers to build relationships and raise awareness of issues is the bedrock of any advocacy campaign. Meetings with the relevant ministers, shadow ministers, advisors, senators, government departments and key backbenchers have taken place to inform and influence government policy and build political support. Regular meetings with the National Disability Insurance Agency were established early on and have been instrumental in contributing to the design and development of the scheme to ensure that the needs of people living with MND were considered. These discussions are important in not only raising awareness of the need for change, but also in showing that the MND community is vocal and working together. In addition to these meetings, MND Australia has organised Global Day and other parliamentary events and attended relevant hearings and roundtables. Crucial to all these activities is the involvement of people living with MND, their carers, family and friends. A key advocacy activity is also writing submissions. And since 2006, 70 submissions have been made to all relevant productivity commissions, Senate and community inquiries and royal commissions. In addition, pre-budget submissions and responding to budget announcements, developing pre-election statements and writing to ministers and shadow ministers are important aspects in effecting change and building political support. Developing national position statements and joint position statements with other peak bodies on a range of issues underpin and guide our advocacy, and also aim to clearly articulate the policy changes required. There it is. Informing and educating the wider community is a vital component of advocacy and key to increasing understanding and awareness of MND. Surveys and research undertaken by MND associations consistently highlights that access to coordinated multidisciplinary care and support from health and community care providers who understand MND. I think I've got this wrong. I'll just checking this. Have I gone too far there? I think I have. I'll get back. We go back. Not letting me go back. First thing on here that doesn't.
You could try the arrow on your keyboard.
Stop doing that.
Oh, okay.
It's all gone a bit frozen, I think. Anyway, that's all right. So I'll talk about personal storytelling, see how that goes. So personal storytelling, it's engaging the community here. It's key to influencing government policy and informing decision makers about the impact of MND on individuals, carers, families, and the community. Wherever possible, case studies are included in submissions to demonstrate the real-life impact of government policy on people living with MND. People with MND, carers and past carers invited to attend and be involved in parliamentary events and meetings with decision makers. And it is these personal perspectives that have made the most impact over the years. The community has also been engaged through mainstream media stories, the annual MND Week calls to action, social media campaigns, the 5 Rights Commission, and awareness campaigns such as the Global Ice Bucket Challenge in 2014 and the Fading Symphony in 2015. The use of social media has grown considerably over the last 15 years, and community engagement through Facebook, Twitter, and Instagram has increasingly provided the opportunity for people to tell their personal stories to amplify advocacy campaigns. Trying again now, not moving at all. Somewhere on the screen I can do that, I wonder.
Try hitting the escape button and going back to your master
Yes,
slide.
I might have to do that, I think. Sorry, people. I think I might be a little bit slow on the bandwidth or something. I might go back to that one. Yes, I think my presentation may have frozen a bit. Whoop. Not the one I wanted. Where is it?
That's it. Double click on that one.
Is it coming up? It's just not coming up as check. Go right back to the beginning. Oh, sorry, everybody. It's not very good, is it? No, I think it's frozen. So we might have to do it without the slides, I'm afraid. It's just coming up with that first one and not moving. Aha. Oh. Hang on in there, we're getting there. Oh.
Carol, try the lower sort of left-hand side of the slide. There's arrows. Try using those.
Yes, I'm not seeing. Oh, there it is up the bottom, down the bottom there. Let's try that one again. Sorry, everybody. I think I must have gone, my internet's gone not very well, I think. All right, well, we'll have to go without, I think, and see if it catches up in the end.
Yeah, just wing it, Carol, you'll be right.
Yeah, I'll just talk. It's coming up with low system resources, so there's obviously an issue somewhere along the line there. So I'll go back to talking about educating and informing the wider community. So as mentioned, that's a vital component of advocacy and key to increasing understanding and awareness of MND. And surveys and research undertaken by MND associations consistently highlights that access to coordinated multidisciplinary care and support for health and community care providers who understand MND is a priority for people living with MND. So over the years, there have been many conferences, meetings, information and online events have been developed and held. And in addition, many more similar events have been held by the state-based association. So engaging with the myriad health professionals and service providers involved in MND care and providing opportunities to inform, educate, and share best practice is vital to influencing and promoting a timely, coordinated, multi and interdisciplinary approach to healthcare and the development of MND specific clinics, services, and models of care. This engagement is also crucial in encouraging and supporting the implementation of evidence-based interventions and promoting health and social care research. A relatively small cohort of 2,100 people living with MND at any given time, rapid progression, and the fact, as David has already mentioned, that MND is classified as a rare disease, has an impact on the power to change government policy. With this in mind, MND Australia has from the outset focused on highlighting the unique challenges of MND whilst partnering with other peak bodies, developing strategic alliances and leveraging community campaigns. Partnering with key national, international organisations to promote and support collaboration is an important activity for MND Australia. Identifying strategic partnerships to effect a specific policy change strengthens submissions and amplifies our collective voices. Collaboration was a key element of the success in influencing the introduction of the NDIS in Australia and will be key to effective changes to aged care and also to research. I'll just see if I can move this on now, whether it's come to life. Anything happening? Oh, no, it's gone to the end. Okay, so I hope this, so to conclude, there's been a clear advocacy focus over the last 15 years with broad and positive impacts. And that's where I wanted the slide, because it's a busy slide with all the impacts on it, which is a shame it's not coming up. Anyway, maybe it'll appear at some point, not yet. So one of the major impacts over this time has been the introduction of the NDIS. Since 2008, MND Australia and the state MND associations worked to influence the development and rollout of the NDIS in recognition of this once-in-a-lifetime opportunity for change. People who are diagnosed with MND when under the age of 65 can now access funded services and supports to meet their needs and can continue to participate in the scheme when they turn 65. Access to assistive technology and health-related supports, including non-invasive ventilation through the NDIS, has also had a major impact on the quality of life of younger people living with MND. Unfortunately, for older people diagnosed with MND, challenges continue. And it is hoped that with sustained advocacy, the changes as outlined in the recommendations of the Royal Commission's final report are implemented in full. There have been a number of impacts related to health and research. Access to multidisciplinary clinics, services and models of care is now available nationally in all capital cities, apart from Darwin, and the ACT, and hopefully Medicare-funded telehealth consultations will increase the reach of these MND-specific services. And as outlined by Karen in her talk, there's a lot of research around this still ongoing. Research funding has increased considerably since 2006, with a consequent acceleration in research discoveries, assistive technologies, and access to clinical trials. So I hope this overview has provided you all with an understanding of MND advocacy in Australia. It's certainly confirmed that influencing government policy is indeed a long-term commitment, requiring focus, flexibility, and a lot of patience. Most importantly, I hope it has highlighted the vital role of people living with MND, their families and friends in advocacy activities, and also in ensuring that MND Australia continues to be focused on and informed and motivated by those impacted by government policies. On the final slide, I did have the current focus and the continuing call to improve equity of access to services and supports for people living with MND, particularly within the aged care system, but also within the NDIS as reviews are underway. That constant keeping on top of what's happening on the NDIS is really important as well. And also obviously in the healthcare sector and the research sector, with around access to genetic services and support as well. But I'll let David talk more about current and future advocacy activities at MND Australia. So thank you, everybody, and sorry for the glitch.
Thank you so much, Carol, for that comprehensive overview of the history of advocacy in MND's advocacy, and yeah, shame about the technology bit. That's the way things are these days. So I'll hand over to Peter, who I introduced earlier. I know he's a great advocate in so many different ways, and I'm sure that he'll probably share some of those mechanisms. So Peter.
Thank you, David. Thank you very much. And everyone got me all right?
Yes, we do.
Nodding. Thank you. Good. Sorry, just clear the throat a bit. Firstly, thank you so much for holding this. I mean, it's really good to get a front row seat to an event like this, because you get a greater understanding of the effort and that does go into the research and all the other issues associated with MND. So yeah, again, thank you for holding such a great initiative, and also for having me as a guest, which it's a great honour to be able to talk at such a forum. I'll open with my brief background. My first symptoms were back in, probably hard to put a real finger on it, but around the 2015, I started to feel numbness in my hands. Doing up buttons became a challenge, and just getting my arms above my head was a bit of a challenge. And that led to some surgery I had on my neck in 2018. And my advice to anyone is when your surgeon comes to see you the next morning and you've lost the use of both your arms, and his jaw hits the ground and says, "Oh, gee, that shouldn't happen," you probably know you're in a bit of strife at that point. And that's how it unfolded for me. And then the process of trying to find out what the cause was took probably another couple of years, 18 months or so. And I was finally diagnosed. And to be honest, it wasn't a relief, and that's a silly thing to say, it was a relief, but in early 2020, I was formally diagnosed with MND. So at least then you can get your head around, what the issues are and okay, where to from here. So that was, as I say, early 2020, and start of a friend has called it my MND career. I retired from work due to I just couldn't work any longer. Sorry, I've got arm flail is my issue, and my arms are pretty much gone now. I can do certain things, but it's requires effort, and they're less and less useful. And as a consequence with work and the fatigue that came with the disease, I finished in, I think it was Easter of 2019 when I finished work. So for a period there, I was just getting my head around it and working through the system. And a mutual friend of mine introduced me to Nick Brax, who's a guy who's an advocate for mental health and works in the mental health space and runs a series of podcasts. And I was a guest on that, and that was the first time I was able to talk my own journey out to myself almost. It's been running around my head for all this time, and this was it's probably late 2020 when I was a guest on the podcast. And yes, as I say, just that ability to dump all that information at once and almost listen to myself as a third person talking about it was really, really valuable for me. And then Nick made a book. He was writing a book at the time and asked me to contribute to that. And so I did so, but I thought, well, I can't type anything. So I'll just talk to my iPad and record it and send that off to Nick that way. And then that became a series of YouTube videos. And I've got someone working with me now who does all the magic behind the scenes. I'm hopeless with all that stuff, but she's putting together these fabulous little videos for me with such great titles as How to Eat a Packet of Chips with MND. That's a bestseller. I implore you to go and have a look. I can see some nodding there, so I've got some some people buying in. And How to Survive Getting punched in the face 100 times a day. That's another important one. So that's given me, again, time to sort of just talk things through in my own head, and then that's, I suppose, been my main form of advocacy, is just getting my message out onto to the community via YouTube. And I've come to know David pretty well, and we speak occasionally, and got a couple of projects sitting in the background, which we're talking about, which is great for me. It gives me a purpose and a reason for, and I suppose a method of tackling the disease, my own little method, rather than, again, I'm dealing with a community who knows what's going on here, and the alternative is roll up in a ball in the corner and feel sorry for yourself. And that's not me, but gee, it'd be so easy to do for some people. And I'm not going to, I understand how some people don't cope with it, but I've been fortunate to sort of find my own little way of coping, which has been great. From a point of view of I suppose my messaging around advocacy, there's probably a few aspects there that I'd like to sort of talk about. And one is you almost need a process map in front of you. I had to go and find so many things, and that was one of the frustrating things that. And I read the Karen's slide before about 19% take up of NIV, which I just find extraordinary. And it was mentioned to me, and I can't remember how it was brought up, but someone mentioned to me early about NIV, and it's only, again, sort of, I get these raised eyebrows every now and again when people, oh, you're on NIV, are you? That's great. Rather than, it should be at the start of the journey, if it's such an important part of the process and life enhancing. So things like that, I could be sitting here now, not without NIV, yet I've been on it for 2 years, and I can feel the benefits of it, because my lung capacity is down about 45%. So there's another challenge. So I've got this ongoing breathlessness, and as a similar issue. So so as I say, a process map almost kind of approach to things where you work through your list of symptoms and issues and confrontations, and you've got the solution there. The solution is available more for you. Again, Karen slided with the trying to find the owl. I'm still looking for that owl, by the way, Karen. I couldn't see it, but anyway, it says more about me than anything else. But that was a great analogy for me to. It struck home that you are trying to. You're not only trying to find that proverbial needle in the haystack, but you haven't got the haystack in front of you. So, it can be an enormously frustrating process to work through. So again, I'm really appreciative of this sort of forum that breaks down those issues and brings them to front of mind, which is really good. And so some of the little tips, and I'd call them sort of ancillary services. Again, I've had to go and find these things, but once you find them, they're terrific. I've just got a list here of things like half price cabs. I'm in Melbourne, so I'm not sure if that's a national initiative or not. The companion card here, which means that my companion can go with me to events, and that's free, which again just breaks down the cost of things and means you can go out into the community and without at a cheaper cost, and just makes that little bit easier. Last couple of years, again, as everyone appreciates, winter's not my friend anymore. So I've had this, I suppose, somewhat of a pleasure to spend the last 2 years. We've gone to Queensland for 3 or 4 months just to get away from the Melbourne winter. So that taught me things about travel and how to best use the facilities that are there for travel. And I'll give you a great little example is the Sunshine Coast Airport. where there's a number, an assistance number, it's a mobile number, it's a guy with a phone. You give him a call 5 minutes out, and he's your arms and legs for that period. And I had the opposite experience at Canberra Airport, where I was basically treated like a terrorist is a bit harsh, but I was certainly treated like a person of interest when I couldn't get my arms from my side. And that was a terrible experience. And thankfully, I was able to speak to the head of security there, and he was incredibly apologetic and supportive of my needs. But it was, after the fact, because it had already happened. So I've seen the two extremes of travel. And little other things. So I want to mention the NDIS, which is such a critical support here. And I understand, again, the issues around equity between under and over 65. And I'm pleased that there's so much work going on with that. But as a user of the NDIS system, I can't speak highly enough of that. You can only really truly measure these things by taking it away. And I know that my life without the NDIS support wouldn't be anywhere near what it is. I'm able to get carers in to just do those daily, the first hour of my day is getting me out of bed, showering me, shaving me, dressing me, cooking me breakfast, all those sorts of things. But I just can't do any more. And it's an unfair burden on my family to have to do that constantly. So I'm very, very grateful for the care that I'm able to get in to help undertake those processes. So really, I'm just, again, I'm referring to notes here, but if my little, contribution can be that we turn this into more of a solutions-based approach, that we're looking for solutions, and the solutions are up front and available, and rather than being a question-based, that you've got to go looking for things. I think the clinicians and everyone involved in the community from a care aspect, if we sort of flip the mindset a little bit to say, we're going to look for, well, here's the solutions. We know the solutions. We haven't got a cure, but we've got a pretty good idea of what people need to enhance that quality of life, and to make life as easy as possible on everyone that you touch, on the person with living with MND, but the critical people around them too, of family, friends, carers, and the like. So yeah, that'd be my final sort of takeaway, to put that one up in lights, that we need the solutions front of mind versus a mindset of asking questions after. That's probably all I've got. So open for questions or comments.
Cool. So if anybody wants to think about some questions for both Carol and Peter, and I just want to thank you, Peter. The unfortunate thing now is that you mentioned about sort of how to eat a packet of chips with MND. So every time I have a packet of chips, I will remember your phrase. I think Peter did mention to me the issue that he had at Canberra Airport. And even though that we're related to Peter solely, it certainly has some much wider issues for other people with MND, as well as other body movement functional challenges. So I actually took his calls up with Canberra Airport, and they were certainly going to raise it more broadly across some airport federation or committee or something or other, to ensure that all Australian airports, both metro and regional, sort of are more attuned to issues like Peter has shared. I've noticed Keith's got his hand up, so I'll ask Keith.
Thanks, David, and thanks to Carol and Peter. Two good presentations, and the chip story I understand fully. In Carol's presentation, the advocacy role was predominantly represented as a series of activities to convince people about the need that other people might have. In the whole presentation, there was a single slide that had the unfortunate dollar sign on it, which was the 2015 thing. my suspicion is that it won't be difficult to convince Bill Shorten and others of his persuasion that we have a problem that needs to be addressed. But what he won't say, nor his friends in the back of his head, is how much is this thing going to cost, and why do these people need it more than another lot of people who are telling me equally about the issues of their particular world. So where does the advocacy function go in relation to the answer to those two questions? I mean, the government has some limited amount of money, already putting a lot to it. Should we get over 65 treatment as MND people more or less than any other mob of people who might want like access when they're over 65? Does the government then have to go fully to the NDIS and disability type people past 65 and never treated in the state aged care? What's the line of approach for that side of it?
Carol?
Oh, thanks, David. Yeah, I guess the line of approach has been to highlight the discrimination, and that has been taken up, as you may remember, by the crossbenchers who've highlighted the discrimination for people with a disability. So the aged care system is designed for the frail aged, and I think that's where the point of difference has to be. That when we're talking about older people with a disability or who are diagnosed with a disability, there is that difference, and those people do need to ensure, we need to ensure that they have access to disability services, whether it's funded through aged care or through a sort of NDIS type supplement approach. That's for the government to think about, but it's certainly part of the Aged Care Royal Commission has identified the fact that people with disability who are over the age of 65 do need additional supports that are focused on their disability. And that's, again, we're partnering with organisations like the Neurological Alliance of Australia, which we have done for many, many years, and David is doing now, to partner with other groups to highlight the impact of the aged care system for people who have a disability who are older, and how the system just doesn't address the needs of people who have a disability. I did meet with Bill Shorten a couple of years ago, and he certainly understands the discrimination, but it does come down to dollars, as you say, Keith. That's the problem.
And just one thing I want to add, Keith, in response to your question, is that Bill Shorten, as well as his colleagues, are very supportive of the challenges that people with MND have. And in all the conversations I've had with ministers and backbenchers and crossbenchers and so on, invariably most of them know of somebody with MND or who's had MND. And so that, that personal connection is really helpful as we're telling our story and trying to influence change. The other thing that we did was we've established the Parliamentary Friends of MND group. So we launched that at the beginning of September. And we have, we initially had 13 members and senators as part of that group. And now we have 28. And of those 28, 8 of them are ministers or assistant ministers. And just to give you a bit of maths, because I had to work it out myself, that 28 members and senators represents 12.3% of all politicians, federal politicians. So why have so many joined MND, the MND Friendship Group? And a lot of it is because of that personal connection, even though it's a small disease or relative to cancers and others, and they want to make a difference. And to have 8 ministers involved will actually help us, through them, influence some quite significant policy and legislative change. Peter, I don't know whether you wanted to add anything to Keith's.
No, I think Carol covered that pretty well, David. But yeah, I mean the issue of the 65 under or overs are, it's a big one. So out of my pay league.
Good. A question from Karen. Casey, sorry. Yeah.
Hi. So just for context, I work in government relations and I live in Canberra, and I used to work up on the hill. So I just have these are sort of questions I wouldn't necessarily very much with the government relations hat on, like these aren't sort of things I would discuss with my mum, for instance, who has MND. So I'm sorry if it sounds insensitive, because that's not the purpose of asking these questions. But when we talk about, for instance, the Deloitte report and that evidence-based policy, not just a lived experience, but that sort of more, we know 2,000 people at one time might have MND, and this is how much it costs. But will that Deloitte report be updated, because it is almost 8 years old? And is it time for a refresh? And would there be an opportunity to also talk about the positive economic flow as opposed to a cost, in terms of, the fact that there are, my mum has all these carers around her, and it's essentially creating employment opportunities. Not that I hate having to link that, but often with ministers and politicians, there is that people to people experience, but then having on the page what that looks like in an economic sense. And with the Parliamentary Friendship Group, interested to know what the plans are and how I can support, even as someone who's sort of heavily linked. And I mean, I have a lobbyist pass and am at Parliament House quite a lot. And I've offered it a couple of times, but probably very from a distance. So I think I've sent an email or spoken to someone who made a phone call to me from one of the other organisations, I think, about how I can support.
Okay, well, thank you very much for that support, and very happy to have you and anybody else involved. Peter, for example, was one of the speakers at the parliamentary event last year and spoke very eloquently and very passionately. So very happy to have anybody and everybody involved in whatever way that sort of best suits everybody's. In regard to your first point around the Deloitte Access Economics report, we're actually in the process of just finalising the scope to update the report in terms of, not only in terms of the dollars from 2015 to 2022, but more importantly around the economic and social impact of MND. And so we're going to take and we're going to weed through. The 2015 report was a very academic report. It was a sort of economic analysis, and this one's going to be more people focused, and so we're going to weave a lot of stories through this particular report. So we've actually got a schedule in regard to approaching a number of people with MND and carers and past carers and others to help with that process. As I said, we're just finalising the scope, and recognising where we're at in the year. It probably would be early next year before those sort of workshops would start. But again, that will give us a very tangible vehicle to go and talk to people and say, here it is. Here's the economic costs, the social costs, and what we're proposing to do is to have some solutions to say, if the NDIA was going to free up streamline their home modification application processes, so people don't have to wait up to 2 years to have a home modification. That the benefit to the NDIA is X, the benefit to the person with MND and their family is Y, but the savings to government is Z. So it's taking a much broader focus. In regard to the Parliamentary Friends Group, very happy to, as I said, to have people involved. We're proposing to be quite different from a number of the other parliamentary groups, which tend to be event focused. We're going to have a couple of events during a year, but it's really about using the group. We've got 28 people who have signed up, and it's about using them as a sounding board. The Deloitte report will be one sounding board, so when we get the sort of the penultimate draft, we will take it to the Parliamentary Friends Group and say, well, what do you think? We'd love your feedback before the report's finalised. And then there'll be some other opportunities, like get them more aware of research and what's happening in that space, and and the various challenges that we've got. So they then become our voice to people like Bill Shorten, Mark Butler, and other ministers.
Who are the main champions at the moment in that backbench cohort?
It's
would you say?
It's it's the group is co-chaired by Alex
Hawke and Carol Brown.
So for those who don't know Alex, he's a Liberal ex-minister from New South Wales, and Carol Brown is a senator, Labor senator from Tasmania. And then, as I said, yeah, there's quite a long group, a long list of people, including people like the Assistant Minister for Health and Aged Care, Jed Carney, the Assistant Minister for Aboriginal Health is there, the Minister for Mental Health, Assistant Minister for Mental Health and Rural Regional Health is a member. So I'm happy to circulate probably through Gethin the list, and he can, so you can actually see.
I think what's really important, because I mean, been around a lot of politicians, there are many who will say, oh, this is a fantastic initiative, this is great. But unless they actually then champion and take that next step of saying, I'm going to own this and push it with my colleagues, and it really doesn't matter. It's a waste of our time, really, as far as I'm concerned.
Yeah, well, I used to also work with lots of politicians, so I get exactly.
You get you get it, yeah.
But I think with a cohort of 28, all we need is 4 or 5 of the 28 to
exactly
to do what. Yeah.
Agreed.
Any other questions for Peter or Carol?
David, probably just on that. I should probably take this offline a bit, but on that parliamentary side, as you know, you mentioned before that I have spoken. I find that, and that day was a classic example of it, and today as well. Whenever I write these sort of speeches in my head for this sort of occasion, and then have to take out the spite and the anger because it's tough work. So if you need any speeches given with the spite and the anger in, if they may be appropriate, then please call me.
No, very happy to, Peter. And I think I'm aware of the time, but I think you're a great group of people who've got that lived experience, who will be fabulous ambassadors to help MND Australia and the state associations in terms of our advocacy efforts. So reach out, whenever you can, and we'll add you to the mix in terms of opportunities. So, thanks, Gethin.
Thank you, David. That was fantastic. Thank you, Peter and Carol as well. So that brings us basically to the end of the workshop. I think, Rick, are you going to just, Rick and Alison, just going to take us through how you see the sort of ambassadors working, or how they've worked in your experience? And I'm sure you identified a few synergies and correlations with what we've been discussing. So please.
Yep, Alison's going to take this part of it.
Okay.
Great. Thank you. I'm assuming you can see my screen. Okay.
Yep, we can. All good.
Yep. Okay. So thanks for letting me spend the last couple of days with you. It's been great. Just like I think Kathy had said, the title, congratulations, let's do this, kind of implies I know exactly what you should do next. And spoiler alert, I do not, because it's kind of a very personal choice. But I've learned so much about where we're different, but also I want to share a little bit about where we're the same, and what being a research ambassador, how we can work together to move things forward. So just very quickly, a little bit about me, because we're all part of this family or this community. I'm super competitive, love to learn. I prefer to eat healthy, other than if there's Mexican food or donuts around. I'm weak. I'm a little bit persistent, and I'm a type A++ personality. Just so you know, I can drive people crazy. And sometimes there are people on this call that have to hear about me a little bit more than they want to. So I always have to have one fun fact. So my fun fact for you guys is I probably will never visit you in Australia. I'm sorry, but the reason is I'm a terrible traveler, And the reason being is I will not go into the bathroom on a plane because I think people have actually been sucked out during the flushing process, but nobody wants to talk about it. So I'm just going to say, I will see you on Zoom. But of all the things I am, I'm a surviving spouse of ALS. This is my late husband, Jeff. We were married for 20 years before ALS came into our life. So I understand that ALS doesn't happen to a person, it happens to families. And once you're impacted by ALS, you're always impacted by ALS. And I believe that there's hope right now to redefine what being a survivor of ALS is. And so that's why I'm here, and I'm really excited to work with you on that. Very quickly, a little bit about NEALS, because NEALS is where you will be a certified research ambassador through. So NEALS is the scientific consortium in the United States and various other areas throughout the world. They're always growing. But it is not an advocacy organization. It's a scientific and education organization, but we do have subcommittees. And one of those subcommittees is the Patient Education and Advocacy Committee, which I co-chair, which is Dr. Bedlack's invention from many, many years ago. But that's where the Clinical Research Learning Institute lives and where we are research ambassadors representing NEALS from. So I'm all about fun facts, just a little bit of fun. We have a lot of research ambassadors. We've trained over 500 research ambassadors in 22, now 23 CRLIs. So we are forever growing our perspectives and kind of reaching outside our borders to see where we can work together. So if I had to say, well, what does it mean to be a research ambassador? Our goal is to keep the conversation going, right? We want the world to know about ALS, but we want to keep the right conversation going. So how do we do that? Like, what do we talk about? Where do we find the information? How do we know that the information is valid? I've heard a lot of people bring up social media today, which I appreciate, because it's really important for my friend Abraham Lincoln here that you don't believe everything you read on the internet, right? There's so much. Social media has opened the door for us to spread information, but you can also spread misinformation like confetti nowadays, right? And it's very difficult to tell what is what. So as research ambassadors, we really work together to make sure that we are understanding what we're reading and spreading good information, always respectfully correcting misinformation and making sure that the right discussions are occurring with the right people. So I think Rick has brought up, and I want to make sure that you have this, and I'm not sure if you get the presentations, but ALS Untangled, a lot of what we talk about, like the science is out there, the press releases are out there, but there's a lot of alternative and off-label treatments that people talk about. And it's really difficult to know if those are real or if we should be spreading information about those. So ALS Untangled is just every like maybe crazy sounding idea you can think of. And it's scientifically reviewed in a consistent way. So we know exactly what we're communicating about. And I definitely, if you haven't been there, I suggest you looking that up. One of the things that's out there that Rick and I have talked about, my little Sherlock Holmes, that's so cute. Some things to watch out for, the red flags when we're looking at information, the lack of safety, no informed consent, lack of evidence, anecdotal evidence only, no real training in science. The worst thing that can happen is people interpreting science for us that have no scientific background, and then we take that as good information and spread that. So we want to be really, really careful about that. So how do we do this? We have all this information. How do we take action with that? And I just want to say, your story matters because you matter. And I think sometimes we're kind of in that line is drawn in the sand where it's people impacted by ALS and people who are not impacted by ALS but are trying to support them. And the truth is, we are them. Like, we were all in the same grocery store line until one of us got that ALS diagnosis, right? So we really focus on, we have a perspective having been impacted by ALS, but we also have mad skills to bring to the table. And our advocacy is important because we can take who we are in our careers and in our life, add that with our perspective and our lived experience, and really create some amazing change. Sorry, I'm talking fast. But as I'm listening to all of these things, I want to ensure you, like, there is some loud and large advocacy that happens, right? And we all love to talk about that. and all these amazing things that we're doing, but it can also happen very discreetly. And you can participate virtually on an individual level. There's all kinds of opportunities to advocate for yourself and for others. If you're not comfortable being in the public eye, that's totally okay. And we can work together to help you achieve your goals. So how do you get your goals? Well, you have to kind of know what your passion is and kind of who you need to work with in order to do it. Just like as the patient community, we don't want the scientific community to duplicate their efforts, right? We want people to work together. Well, we kind of have to do the same thing. And if you're passionate about something that already exists, even if it exists in the United States, come on over, like experience it with us, be a part of us, and take it back and modify it to work for you. We're oh so willing to do that, happy to do that. So how do you think about this? Think about you and I am and what those words mean to you, and break it down really simply. Like, this is what I want to do. These are my goals. Make a list. Everybody loves crossing things off a list, right? Sometimes I like write on my list, make a list so I can cross that off. And yes, good job. But make a list, do your goals, and find somebody who's going to be your support person. And I am 100% serious when I tell you, if you need a support person, no matter what time of the day or night it is for us, because we're on different sides of the world, I'll be there for you. Email me, what have you. The crazier your idea, the better. Maybe we can actually make it happen. There's lots of crazy ideas that are actually coming to life. So now you have completed this course. You are considered a certified and vetted research ambassador for the NEALS Consortium. And as a default, you become part of that subcommittee I told you, the Patient Education and Advocacy Committee. And what that committee does is we bring all the players to the table. So pharma, anybody in the industry, advocacy organizations, scientific people, researchers, clinicians, we come together once a month. And I'm going to be honest, right now, you'd have to have insomnia to attend that because our timing is not going to work for you, but we're addressing that. We're working on it, whether it means that we're recording it or however we do it so that you can be involved. You are welcome at every one of those meetings. You are welcome to participate in any project that's presented for people to get involved in with any of our companies. So our next meeting is November 14th at noon Eastern. So if you're awake and you don't know what to do, I will send you that link so that you can be a part of us, but you will automatically get the meeting minutes and anything that we send out after, now that you're officially a research ambassador for NEALS. So I just want to say, I have some other screens that have a ton of resources for you. Hopefully, Gethin, you can share those with people, but so we won't go through any of that, but I just want to encourage you to live your life from your heart and share your story. It's very healing. You matter. We're all in this together, and this isn't the end. This is the beginning of something really wonderful. And one of your first advocacy assignments can be to help us in the United States come up with a partnership that makes us all be able to work together. So I appreciate everything. That's our contact information, which we'll hand out as well. Please feel free to contact us at any time, and I will turn it over to you, Rick.
Thank you, Allison. I want to thank everybody for participating. It's been a fantastic 2 days, and I'll just leave you with a couple of my favorite quotes. One is from a person named Margaret Mead. She said, "Never doubt that a small group of thoughtful, committed citizens can work together to make a difference." In fact, that's really the only thing that ever does. And now you're part of that small group of research ambassadors, and I know you're going to go out there and make a difference. And the second thing I'll say is, you know, I've heard about some problems that I didn't know existed in your country, but I've also heard about some possible solutions. And I know it can be daunting to try to take on things like disparities in care, disparities in accessibility, you know, availability of research options, availability of medications that are getting approved in other places. But someone else once said, I think it was Buddha actually, the journey of 1,000 miles, it starts with one step. You all have unbelievable stories, passion, charisma, and now you're empowered with knowledge of research and a network of people in Australia and over here in the United States who will try to help you with your own unique missions. Go out there and take that first step. I can't wait to see where your journey leads. Take care.
Thank you, Richard.
Thank you, Rick. And well, I'd like to say a huge thank you to all, to yourself, Alison and Terry from Niels. It's been awesome having you along and really guiding us through this. And, you know, God knows what time it is for you. It's stupid o'clock. And so we really, really appreciate that. And look, I've got a few thank yous I'd like to say, obviously to all our speakers. They've been awesome from both across the 2 days. And I think, as Kathy said, they're a very friendly crowd and they are utterly devoted. When I reached out to everyone to say, would you mind presenting over this, giving up a couple of days of your time, doing it on the Saturday, they snapped my hand off. It was like no bribery, no coercion needed. Everyone just wanted to jump in and contribute. So it just shows really how keen. And I can tell you from the emails they get on weekends and late at night that they work hard. And look, I also like to say a big thank you to Genius as well for partnering on this. They really kicked us into gear to get this moving as well. I've been talking about this for a couple of years, and really, I mean, I spoke to Rick about it, I don't know, 2 or 3 years ago, and it wasn't until Genius came and really put it right to the front. Let's run this, and I think hopefully you'll agree it's been a thoroughly wonderful experience. We've all learned heaps. I live in research and I still learn a massive amount, and it was great to meet everyone here. I think we've got a really good grounding for an Australian ALS MND ambassador network, and I'm hoping when we keep running this, it's just going to keep getting bigger and better. And we've obviously got a lot of things to be getting on with in the advocacy space, and I'm hoping everyone here is going to be leading the charge a bit on that as well. And look, that's my biggest thank you. I mean, I thank the speakers, thank the guys from the US, Genius, but really there's no point running this unless we have all of you with that lived experience, you know, and to be engaged and willing to help your community. And that's really critical, you know, and that's what's beautiful about the MND ALS community, that everyone does realize that you're working for a bigger thing, and it's not just about you, it's what you can do for, unfortunately, people following or and who's still struggling with this. So that's what I think we can do. That's all I really want to say. I think you've listened to me enough over the last couple of days, and I'm sure you want to get on and enjoy the rest of your weekend, or start your weekend for our US colleagues. You can have a good night's sleep and then it's Saturday for you. That's it from me. Does anyone else want to say anything before we sign off? Other than that, then it's been great meeting you, and you will be hearing from us. We'll be sending some information around with a bit of a survey, all the links from Niels as well, from Alison. We'll send around all those extra information, and the slides and a whole bunch of stuff will be coming your way as well. So looking forward to seeing you many more times, and enjoy the rest of your weekend. Good afternoon.
Bye, everyone.
See you.
Bye, everyone.
Thank you, everyone. See ya.
Bye.